whatever fieldwork we do is carefully curated and preserved for the long term, because it may be the only time you ever get to collect that particular sample in that particular environment. Does that make sense?
Yeah, thank you very much. That makes sense.
Okay, thank you so much. Thank you so much. We can go on the... Yeah. Okay. Yeah, thank you. Okay, thank you so much. Because of time, we have still the technical sessions to take. Let's hear from our discussers. So my first question will go to our distinguished fellow, Professor Moody. Most of our hear from our discussers. So my first question will go to most of our producing compounds from non-plant sources, marine, fungi, and the microorganisms, have not found their way to clinical use. So what do you think are the barriers, preventing them from being used or being available as viable drugs? And what do you think we can do to better bridge research, policy, and industry in order to make these compounds available as therapeutics?
Thank you very much for that question. Sorry, I'm trying to... I think the lead speaker has actually spoken about this. I think one of the major problems, although people say we cannot talk about that, but you can see, for example, South Africa having facilities for really doing this work, especially the marine area and things like that. We, in many places in Africa, we still lack these resources. And I think that is the one thing that needs to be addressed. Secondly, I have little issues with this topic anyway, because I don't think, well, in my own little biology when I was growing up, we have this fungi, we consider them as plants. So I think maybe what we want to talk about this topic is non-higher plants. Because when you talk about bacteria from the soil, from the marine, we consider them as plants, except abroad. That's what we used to think. That, I mean, these are of course plants, but are non-higher plants. And in that wise, we may not all talk about marine, but even the soils that we have, the different ecosystems in Africa, these are areas we need to harness. And as she has demonstrated, we can get soil samples from different ecosystems and get, fungi, the bacteria, culture them, and translate them. Now, this is being done elsewhere. But I think the capacity, the policy, and the will to do it is what is still lacking. I worked, for example, in a little postdoc in the Zenova company, which goes in the UK, which goes all over the world to carry soil samples, bring them and then culture them. And they have arrays of acids to put them through. And within 2-3 weeks, they're already going area to patent. So I think it's just a will for us, as she demonstrated, that it is quite possible. I think, and being in different zones, I think one of the major problems we have is just working together, really having capacity to have a consortium and work together and get these, these molecules biases, toxicity, and then of course translating them into real products which can be used. So it still goes on to policy of government and the will of the researchers to work together. But I think, as she said, we have a national repository center, which can be a good start for us here in Africa. Many times we don't work together. Most of our compounds go out of Africa to Europe, where within minutes, within weeks, they can also be translated to something else. Thank you very much.
Thank you very much, sir. I just want to say, okay, she wants to say something. Yeah. She wants to say something.
All the time, right? But the reality is, our little town, until very recently, we've had massive power outages where we've been load shedding. We call it load shedding, where we have rolling blackouts because our power grid was problems on services and water. And I started with a lab. I didn't have a lab 5 years. My lab was only finished. We had massive power outages when we load shedding. We call it load shedding when we had a blackout because our power grid was and water. And I started with a lab. I didn't have a lab. My lab was only So the thing here is, you don't. The marine natural products work is difficult because you need boats and you need expensive equipment. And I'm just very lucky to be in a town with an institute across the road that has that equipment and that we are sharing. But in your country and in West Africa, for example, the soil, the microbiology in your environment is almost entirely unexplored, and one doesn't need sophisticated equipment to do the bioassays to make the first round of extracts. You know, we have a saying here: one eats an elephant one bite at a time. And the trick is to look to see who has what capacity, where is there a good electricity supply, or where are there backup generators or solar panels to ensure. And in the event that there is a power outage, what are you going to do? And we have plans in place that if there's a power outage, we know what we're going to do. So don't let the enormity of trying to get it all done in one go put you off. I think the idea would be to start small. We have undergraduate projects where our students go out and collect soil samples, bring them in, characterize them as part of their undergraduate practicals, and then those samples can get curated into a collection. The most important thing is, whatever you collect, one needs meticulous records, where it was collected, GPS coordinates, and the proper permitting, because if in our country we don't have permits, we can do nothing, and we cannot take anything out of the lab. So it's important that whatever is collected is properly curated, and that there is a plan to preserve it, so that those cultures or plant samples, etc., and their DNA, their precious DNA, can be preserved in the long term, so that the next generation of people can work on it. It's one step at a time, and I would be very happy to work with any groups that are interested in trying to put together a consortium to start looking at building that capacity. We would be happy to help with protocols for the screening protocols for the antimicrobial resistant that can be done in a normal microbiology lab. And we have a number of protocols, and when it comes to expensive infrastructure, we are already collaborating with colleagues in Ghana and the Cameroon to analyze, to use our mass spectrometry facilities to analyze their samples. So there are always ways in which one can collaborate to make up for the lack of infrastructure. It's about identifying where you can start and what you can do with the existing equipment.
Thank you. Hello, thank you. So thank you so much for that insightful comments. Thank you, Prof, for your insightful comments. I want us to go on to the next discussant by my right, Professor Olutayo. All right, we have seen that we have current technologies, so many of them, the omics, the AI, screening and the metabolic engineering. All these things are new technologies to enhance our discoveries from natural products. And this time around, we're talking about non-plant sources. So having said that, I want to know how can we, or what do we think is the future of non-plant source research in the area of drug discovery with respect to these new technologies? So what is the future like with our current trends and the current technologies we have? What is the future with respect to the non-plant sources drug research?
All right, thank you. And I want to say thank you to our speaker for the presentation. Before I answer your question, I'll just share a personal experience. Last year, a group of us, researchers, and one of our old students from Obafemi University, now is on his PhD in South Korea. So we planned a project around these marine organisms, trying to, the project was actually on getting phytochemicals from the marine source. So we came together as a group and we tried to put together a proposal for NRF, the TED Fund NRF. So in the course of developing the proposal, we discovered that what the candidate in South Korea was able to gather easily was a big deal for us. So we had to contact the Institute of Oceanography and Marine Photography in Lagos, and they told us that yes, they have equipment that we could use to harvest some of this soil from the deep ocean. So we got somebody from that angle. So we went around and built a research team. We didn't get that grant, unfortunately. But the point I'm trying to make is that researchers need to actually reach out, launch out. There are so many facilities out there that we do not know exist. Not until we started that proposal, I did not know, that a lot of us did not know that there was an institute like that responsible for collecting samples in the deep ocean. In fact, at some point we had to reach out to divers, local divers, to help us collect samples until we got that information. So the point I'm making is, we are young, a lot of us are still very young and vibrant, and there are lots of beautiful tools, technology out there for us to achieve whatever we set our minds to achieve. With these new tools,
hello, please, we are not hearing what you are saying. We can't hear you. Kindly put on the phone.
So can you hear us now? Can you hear me now? Can you hear us now? Hello? Please, somebody should respond. Online participant, can you hear us now? Okay, I'll just one. So artificial intelligence is here to stay, and that is where the whole world is going towards now. We have our basic research methodologies, we have our basic research plans and patterns. But as researchers, we are supposed to, let me use the word, we are supposed to upgrade and do research in the way it is done all over the world. So I'll say that there's a beautiful future for research in this untapped area, because this is relatively new around here, especially in the and we still have a whole lot of biomass, a whole lot of population, marine population that we have not tapped into. So with the new technology, with new procedures and methodologies out there, we can do a lot. And this started with, of course, let's take in silico studies, for example. We all started with crude extractions. Most of us, when we did our master's, our PhD, we had to isolate compounds. Now, the reverse is the case. With in silico studies, we are able to get compounds, active compounds in silico, and then we are able to now go back to the lab and test those compounds for bioactivity. So that makes our work easier, and then we use less materials. So that is where the world is going, and I believe that there's a bright future for scientists in the area of artificial intelligence and new technology that is out there. Thank you.
Thank you, Prof, for that insight. Really, okay. Our distinguished fellow and daddy wants to talk.
Well, normally I would have said anything, but only one thing spurred me to want to talk. I think I don't want us to be going on the same road. I think the issue of not knowing that some institutes exist in Nigeria is what made me to want to say something. There are many institutes in Nigeria that are expected to assist you. The problem is that Nigeria is in all of us. Immediately, institutes are established. Their directors go their own way. And I think it's time we start telling ourselves the truth, instead of glossing over it. I mentioned one in my speech, this issue of oceanography research. My PhD was to start that work. I worked on tulicates. The lady is working on only sponges. There are many organisms in the ocean that are not sponges, that are not tulicates, and other times there are corals and so on. Many of them are even of animal origin. But the issue is not that we don't have the ocean. The problem is that those institutes have been a director of an institute, or research institute in Nigeria. I've been a member of the board, and I know what happens there. The problem we have is that the directors of those institutes, immediately they get there, want to go for glory. Me alone. And instead of reaching out to the public, to universities, we are on the wrong end. Unfortunately for us, we are the ones people see. What is our professor doing in the university? What is our DG? Nobody talks about the institutes. I have never seen anything in the paper anywhere that they accuse the director of an institute of not fulfilling its mandate. I have never seen any organization in our political sphere accusing directors of what have been achieved in a year after spending billions, and I mean billions, And it is only academics that come around every time to talk about progress in research with your limited resources or no resources. Those that have been given millions of research funds don't even bother to look for foreign funds. You are the one running for collaboration, this and that, mostly outside. You complain, the ministry people take it up. They will send up, you complain, they take it up and set up an institute to assist you. If any institute is set up, they appoint a wrong director, and the director wants to become a professor overnight, wants to do this overnight without doing anything, and immediately you see it, it does nothing related to you at all, or getting you involved. I know many directors have fought me, and I've talked to so many of them directly, that they should go back to the mandates that established them. If they are doing their work, your work will be easy. Oceanographic Research Institute has been there for a long time, longer than so many of our universities. It's not a new institution. For what have they achieved? Go there tomorrow. What they told you they can do, you set up an experiment to see what you face. They are busy collecting toes from trawlers that bring fish in. They are not interested in research. That was not their mandate. Go to Federal Industrial Research Institute, Oshodi, tomorrow. They are trying to develop with you technologies for the public. What are they doing? The answer is in the here. Go to even the one that is related to you, Nigerian. What have they been doing? Nothing. What can you assess them? Go to Nigerian Science Village, Shega. Can you assess them? Go to NMDA in Lagos. What are they doing? Can you assess them? Go to Biotechnology Research Institute in Abuja or wherever. Can you talk to them? Do you know have they even marketed themselves to you? That they can get something from what you are doing? I can go on and on organizations by government receiving billions every year and not integrating with what anybody is doing. So we are always back to this discussion. What is the research budget of a university that will fund you going on marine exploration? You have to ask yourself the question. Unfortunately, I've also been a vice chancellor. I know what the budget of vice chancellors are. I've been a deputy vice chancellor. I've been in NUC. I've been everywhere academics can take anybody in this room. So I'm telling you the truth. We have to, as an organization, start trying to influence what is happening in governance and start asking the right question and start directing our politicians to look at research institutes. What are they doing? I wouldn't go to what I know, too much of what they are doing, but I think even if I say anything, it's not going to change much, except we collectively start asking them exactly what are they doing in their mandate. I think some of our sessions should actually be to review their mandates and discuss their mandates. How does it affect us? How do we make sure that they do what they are expected to do? All this charade has been going on for too long. And we all keep on coming back to say the same thing. We listen to South Africa now. It was easy for her to get somebody to assist her. Out of the blues, they were storing the ocean. We wanted to do the same thing long before she even dreamt or dreamt of going on oceanographic research. We couldn't do it. People wanted to collaborate with us. We couldn't do it, even when we have money, because of these political problems. And I think associations like us and many other organizations will have to start looking at organizations set up by government and tell ourselves, let us also shout about them, that they are not doing their mandate. Let us review their mandates. Let us review their budgets. Some of them get up to 70 billion naira in budget to do research. And I'm telling you, and at the end of the day, everybody keeps quiet. We start looking at somebody stealing money for buying land, somebody stealing money for... We are looking at the wrong thing. So the issue is that we have to start trying to be more practical. And the time is for those of us, people like me. I think my time is going, I'm retired. And the last time I had to fight one of the directors also. They know I'm retired. They know I don't have power again. If I line up at the retirement benefit center, I will probably be dead before they pay me. But, but you are still young, and you still have the energy. And that's why those of us here, who are here, who knows a little bit, should talk. Our focus should change. We must, at a session like this, interrogate those institutes that are meant to help us do research, to help us provide something for research. All other European countries take research as a gold mine. If you have all the resources in the world, and people are just digging up your gold every day, at the end of the day it will finish. But if you have a knowledge of something, you will get more gold, more things, worth more than gold from that knowledge. And that is why most countries rely on knowledge. But unfortunately, we don't want knowledge. We want quick money. And we want everybody, even researchers. I can tell you some funny stories about some of those institutes that you see on the... And at the end of the day, they have equipment. I have an institute that bought an HPLC, brand new, Agilent, for long before you have it, for over 10 years, it is still in the rubber. Even to remove the rubber, it's still there, 10 years after. So even what to assemble was a problem. And there were institutes that were supposed to help you. So I think we, as a society, must go beyond a little bit. Be thinker, soldier, and spy. Let us find a way of start interrogating all these research institutes. There is no money that will be given to you in your university that can be the same thing given to a research institute to do the same work. What you can get as an individual, as a lab, will only be a minuscule of a fraction of what they have to use. Right? And I think that by the time we start shouting, then even if we can't force them, we'll be like the story of the tortoise and the lion. Even though the tortoise knows he cannot find the lion much, he will go to the lion's house every day and be shouting. And people will be asking, what is the problem with this tortoise? And that's what, okay, let us listen to this tortoise once. And eventually tortoise was able to marry the daughter of a lion. I think the what I'm trying to tell us is that we must look at ourselves. We keep on looking at outside that they will help us. What will help us is here with us. But unfortunately, we keep on blaming government. Government has done its own. They have set up the institutes. We asked for Central Science Institute. They set up Science Shared Village. What are they going to do next? We asked for Standards Organization. They set up some. You ask for pharmaceutical regulation. They set up 9 bridge. NAFDAC is still regulatory. NAFDAC is not research. You set up, you ask for natural medicine, they set up an NDA. What else do you want government to do? It is those of us who are in organizations that are, even the medical, medical research institutes, what are they really doing? It is those of us who are in this area that must shout, investigate these things, and this is the democracy. They have done what you have asked them to do politically. But we also must also now, as associations, regulate ourselves and start shouting. Look at the mandate. Set up committees to look at the mandate of all the organizations, biotechnology institution, center in Nigeria. Many of them, at least things that are got to do with natural products cannot be less than 7. And I have worked, like he did, I worked in such centers in France. Some of my PhD students went to France to work in that center.
Also derail. But I know that one day some of us that are nice, some people that are okay, that have passion for what we are doing, we find their way to that positions. Thank you, Daddy, once again. So we don't have time. I just have 2 more questions that will end this plenary session. And that question will go to our daddy here, Professor Moody. We have talked about collaboration. Our speaker for today said collaborative programs to be created where we incorporate, involve people, researchers from different universities with different skills. Like when we talk about researchers with different skills and respect to the topic of today, we can look at the chemist, the pharmacist, the marine biologist, and the microbiologist and some other related fields. So that is where it is very important, partnership as far as even regional, regional, so that we can have a drug discovery consortium and the rest of them. So what I want to ask is, what are the practical steps? What do you think are the practical steps that can help in this area of collaboration to help us move, forward in the area of drug discovery from natural products?
Thank you very much. That's a very, very important question. And I think that is where we still have a lot of issues going forward in terms of development in this area of natural products is because there's not enough partnership, collaboration, and effective cooperation among the researchers and developers. And one of the major problems that has been highlighted is that people work in silos, not necessarily networking. So I think the way to go is here, like a conference where you can actually network, and also take into cognizance all other areas, because these days now, all the disciplines are overlapping. You know, you cannot do anything alone. So there must be that mindset of collaborating and getting things done. Unfortunately, in Nigeria, as Prof. Destin has just mentioned, that is an issue. Many of these things, many of these institutes are set up so that they can actually coordinate, so that people don't go, because you find many times some plans that you are working in Benin, another person is working on it maybe in Musuka, and they don't even know, they don't even talk to each other. Sometimes even within the same faculty, I've experienced it, people working on the same plan that are working on, and are not even talking together, they are not even working together. So you just rediscovering the wheel all over the time. So there is a need really for this proper networking, and that is what this thing is all about. This work, at the end of the day, you find out that it's not just for promotion. There are many things that that attends to you apart from just being promoted in the university, which we are not, which many times we are losing cognizance of, and that is why development has been so slow. The other point I want to make is that, you know, when you are trying to develop something, at the end of the day, there's also the need to put in place what we call intellectual property policies, so that nobody at the end of the day feels cheated. People are afraid, traditional medical practitioners, for example, are afraid to pour out their hearts, to give out what they know. Even people in research teams, I've seen research teams disintegrate because of lack of trust. So there must be ad initio when you are going forward, at least to have in mind something, at least areas of real collaboration. There must be proper documentations as to what is going to happen if things go, well and commercial outcomes. So in a nutshell, what Professor just said is still very, very important. Government, in where things work, government drive these things. There are, for example, the Minister of Health is, Minister of Science, they are supposed to drive many things where people can come together really properly networking and doing things together. There are themes, scientific themes, there are scientific projects, there are goals, so that everybody can be working towards that goal. And I think that is what NSB should try to do, as well, showing what we are doing, for example, in different times. There are some things we can sometimes have research topics or research conferences which will be based on maybe specific topic, where everybody, so maybe sub-zona, maybe whatever, where people in that area can come together, can be put together to do work and go to work, not just research but also transactional issues to products. So those are my sort of advice that going forward, number one, we must have that goals already set, then we have conference around it, we can network, and then also bring government into it, as Professor Sen said. We need to really shout and get them together. It's a real problem, but a journey of 1,000 miles start with just one step. Thank you.
Thank you so much, Prof, for that insight, really. There needs to work for us to work together as a team for a common purpose. And I gathered one particular thing from you, that we should trust each other. That trust is what normally, is, for me, is key to our not being together. You be in your department, the same faculty, people are working differently, hiding what they're doing, hiding where they're traveling to, hiding, everything is hiding, solo. If you work alone, you don't go far, right? So this is key, and we should try to trust each other so that teamwork, collaboration can work perfectly well. So my last question will go to Professor Lotai. Yeah. So we are talking about exploring the sources we have. This time around, I'll bring in both the plant and non-plant sources. So resources, we are exploring them, you know, trying to figure out, get one thing to benefit us. But my question is, how do we ensure that these things are done sustainably and ethically, so that at the end it will be a win-win thing? We, the researcher, the society, and the environment will benefit. So how do we do these things to ensure that sustainability and ethical prospecting is key to our researches?
So if I get your question well, how do we sustain exploration of both? So how to sustain it? Well, I remember at one of our NSP conferences, I think about the last two, the society proposed to set up a form of a committee, sub-regional committees, to work on the cultivation of most of our herbals. And so that's where I'm going to start from. If we continue to explore and exploit plant materials, natural products, both plants and animal and other sources, if we don't replace them, especially our plants, we are at the risk of extinction, or getting some of them going into extinction. So I would say, as medicinal plant scientists, as researchers who work on medicinal plants, as we are taking these plants, we should also think of replacing them. That is on one side. On the other side, to be sure that we are even getting the correct plant materials. I was there yesterday when somebody mentioned the issue. Okay, the presenter yesterday mentioned the issue of collecting the right plants. It is very important for us that at the beginning of our research, we get the right plants. From my personal experience, I was working on a particular plant when I started my PhD. Along the line, I went to frame for identification of the plants, and I got a different name entirely. I was grateful because I hadn't gone too far. And so when we have the correct plant names, the correct plant sources, they were able to do the correct thing. Now, cultivation is also very, very important. If we have the opportunity of cultivating the plants that we use for our researches, it will help us, and then we'll be able to, it will also help us with reproducibility. Because most of the time, that's a major problem when we talk about standardization of herbal remedies and herbal products. So we will continue to cultivate these plants, and then we continue to work on medicinal plants as researchers. So it is very important to make sure we sustain medicinal plants through cultivation.
Thank you so much, Professor Lotaya, for this your insight. Really, that is very crucial. As we are exploring, we also think about going back the next day. Because if you harvest, you exhaust. It means an end of a research. And we believe our research should be something that is a continuum. You continue to explore and you continue to have it, because once you start off with a product, you believe that you are improving, and the source will also be intact. So it's very important, as we are doing that, we think about replacement, cultivation, and a lot of other things that are associated with it. So today we have seen the need and what is important, what we need to do, where we need to ask questions. We are told these are the things that are here and here. We have different institutes that can help us. It is left for us to ask questions. We need to also look inwards into non-plant sources, as we have great future, and they definitely we are looking at them as the new road or new path to new generation therapeutics. And the key other thing we gathered this today is collaboration, that we need to collaborate, come together as one, so that there will be a better output from our researches. On these notes, I want to thank all of us once again for this great session, for the time we have spent, and I know that we have learned a lot, and I also wish us well in the remaining technical sessions of the day, as I believe it will end us successfully. Thank you, everyone. And let me thank my co-discussants, I said also, and the great speaker of the day. I don't know if she's still online. God bless her for us in Jesus' name. Thank you, sirs.
Let's give them a round of applause. Thank you very much. I hope we have learned a lot today. Okay, if you look at our agenda, we are still far behind time. I think we'll skip company presentation. We're going into the general scientific section. Well, give us a minute or two to just stretch our bodies. But I'll also remind you that our restroom is still by the right side, by my right. We have our restroom there in case you want to make use of the restroom. So we'll give a 2 minutes break, then we'll go into the general scientific section.
I think they said their mind that they drink water. Yes. Okay, Abel. Okay, he dropped us and I think he went to do something. He wants to see him. He's coming. He will come back. He will still carry us back. I don't know. Dr. Abel. So he comes for examination. That's what I wanted to see. Okay. Okay. Yes, his face is here. News, eh? Very good. You know that when he was carrying me back, he was playing the one this Nigerian president. He said, like, is this radio or is it stage? He said, it's WhatsApp. It's like, they sent to me. He's always carrying news. You don't have to say news. Because the news. Don't worry, he'll come back. Because he's carrying us.
Thank you very much.
Yeah, appreciate it.
Good day, everyone.
Yes, I'm looking for the
Okay, see him. See him. That's why I'm requesting to get us before the format of storage. Supposed to have printed a copy.
No, they don't have it.
We just develop it
Yourself?
Okay.
Professor Moody.
Okay.
Then it's copies.
You say that you're a student from
Oh, when we're doing poster.
Well, they should school past and come back. Yes.
No. We can.
Oh, yes. Okay, he's okay.
She's a good.
Okay, so
Yes, she's there. He's a guru in assessment. We have to be. Chairman. Are you staying there or you stay there? Ma, are you going to stay there? So supposed to stay there.
Let's go there. Let's go there. Let's go there.
Doctor, yeah, I have the protocol there. Okay, let's get it.
Good day, everyone, and a warm welcome to the 35th AGM and 18th Scientific Conference.
So, introduction. Yes, that's all that.
Okay, virtual is ready. We are going to remove our parents from it.
Okay, it's virtual. And that will just be optional. So, we are going to have it spread them.
That's okay.
So that they look corporate now, just look at that and come in. Okay, let's make this question. That's question and answer. That's question and answer. Now for people coming physically, this should be utility. Yes. For physical.
Sorry, for online, yes, yes. For virtual, this should be 20. For physical,
Okay, ladies and gentlemen, we want to go into the virtual presentation, oral virtual presentation. So I want all of us to be seated and then listen to the presenters. They'll be also online to answer questions. We have assessors here to assess them and will give us the result at the end of the presentation. Thank you very much.
the others.
Good day, everyone, and a warm welcome.
We will want to know who is presenting and if that person is online. If that person is not online, please don't play the video. The person needs to be online to answer questions. Okay, good. Ata is there.
Yes, Ambula.
Wait.
Is it the Ata that is presenting?
No. Excuse me, ma'am. This ma'am.
She's part of it.
Yes, she's part of it.
Yes. Let me get another one. Look at this one. Because I'm looking.
Just wait, let's replicate for another person. Maybe
Let's go. Prof has stepped out. Let's wait.
Okay.
Professor Moody is one of the assistants. Let me eat the fruit of my labor.
Sure. Right.
Why don't you sit together?
So explain to her too. How about sit
True.
with you? I know that was listening with you.
Giraffe.
I'm not dismissing you. No, I just came to you.
No giraffe. Your name and number.
So,
Good day, everyone, and a warm welcome to the 35th AGM and 18th Scientific Conference of the Nigerian Society of Pharmacognosy. I am Alfred Atta from the University of Ilorin, Nigeria, and I am Alfred Atta from the University of Ilorin, Nigeria, and I am presenting my team's research work titled Genotypic and Phenotypic Discovery-Based Characterization of valine-rich knotted antioxidant peptides with anti-cycling activity from Carjanus cardia. While peptides and proteins occur in all domains of life, however, the gene-encoded cysteine-rich antimicrobial peptides, such as the defensins, the thioneins, the nortins, the protease inhibitors, and cyclotides, are limited to a selected members of few plant families. Many of these host plants are used in traditional medicine. A typical example is the peptide calatabi-1 expressed in the traditional African plant Odlandia affinis. Calatabi-1 analog, known as T20K, is currently in clinical trials for multiple sclerosis. This illustrates the importance of traditional medicine, ethnopharmacology, which could lead to clinical application. Our study plant, Pigeon Pea, is similarly used in folklore for its anti-cycling property with special interest in the seeds.
I don't seem to understand.
However, the seed of Carjanus cardia needs to be standardized. This is because several ascensions of the seed exist in Nigeria. In addition, amino acid phenylalanine has narrowly been implicated for the anti-cycling property of the seed. Nature-derived peptides such as the cysteine nortins have not been reported in this plant. Meanwhile, an anti-cycling nutritional supplement known as cyclavite is currently in commerce obtained from the seed. So, generating a more scientific evidence, a further scientific evidence, will promote the commercial value of this product, particularly at the international market. Our work was therefore aimed at studying the possible cysteine-rich peptide profile using both bioinformatics as well as lab-based chemical analysis for discovery of novel peptides and drug candidates that may show anti-cycling potentials. We are also interested in the expression profile in the various ascensions. The chemical analysis of the various ascensions showed variations in the expression pattern of both the polar compound and the knotted peptides expressed in the seeds. For instance, the chemical profile for TCC6 and TCC11 look alike, while TCC14 and TCC12 are also similar but presented a completely different profile from that of TCC6 and TCC11. TCC6 and TCC11 appear to be very poor in the peptide expression pattern, but good enough in the polar compound, while TCC12 and TCC14 show a higher peptide expression profile, and I want to also add that the TCC8111 shown in the next slide also showed a very good peptide expression profile for polar compounds. So the TCC8111 presents a poor profile for polar metabolite and may not be commercially useful if needed for its polar metabolites. So the most abundant peptides were isolated and sequenced de novo, as shown in this HPLC profile for these three peptides. So partial sequences obtained from chemical analysis were used to mine the full peptide sequences from the available genome of PGP. Further genotypic analysis led to the elucidation of the full sequences of three most abundant peptides that are of chemotaxonomic relevance. Their in silico toxicity, activity, and structural modeling were obtained. The in vitro antioxidant activity of crude and semi-pure peptide-rich extract of pigeon pea showed interesting metal-ion chelating activity, hydrogen radical scavenging activity, as well as ferric-reducing antioxidant power, as shown in this table. The peptide-enriched fraction, which is the 80% peptide fraction of Cajanus cajan, demonstrated or showed a better sickle cell reversal potentials compared to the crude extract, as well as hydroxyurea, the standard drug used. This is indicative of the potential anti-sickling property of these novel peptides, which didn't show activity in the polymerization experiment, data not shown here, but are producing a fantastic antioxidant property. This means that these peptides may be acting via antioxidant mechanism. And so, in conclusion, we report for the first time valine-rich antioxidant peptides in a seed of pigeon pea, which are of chemotaxonomic importance and relevance. We also report that ascensions of the seeds of Cajanus cajan exist that accumulate different amounts of these therapeutic peptides and other important secondary compounds. I want to thank you for listening.
Hello everyone, good.
Let them drink water. I have dirty water in my room. I said I need Please, can you get to Okay, give me one. We're supposed to ask him this. is going to give you the question. But that time, you should give us time to ask questions. Because the other person is seated. You should ask questions. Okay, it's okay there. Yes. The person is not there. The person is online and listening to the record. And we should ask him questions now. Yes, ask him questions.
But that's more important.
Why the person should be there? So why we say don't play the person not online. Tell the person that we know there's questions we ask. Let me take up my responsibility and be put on the
Just maybe the questions to clear.
Yes. So when you put up, call that person, Dr. Fred, attack, Fred, attack. There. Is it that it's appreciating them? Or if that was the standard of mention?
place you now joking with you. So, on the Facebook, he said, I have to pay that. You just want to commit suicide. So, they reject. So, it's when we have begging you, you commit suicide. You say, you will commit suicide. So, it's free. The friend now told me, can you come to my house? He said, no, you know, just want to commit suicide. So, his friend now told me, post it on their own platform. See what
these things can cause wahala. My daughter, she was that first one that okay, women so there's one that like people currency something.
Okay, mind me.
She was that. So she invested her money. They need to say that she had. The mistake she made is that she was the first person that joined it and was like, the money was just trickling. She now involved others. And now served as a mediator. So allow them to cook. They collected money.
You know, they will tell you something if you bring them.
My daughter was and that one day, okay, also her like, okay, bro, she's just this kind of drag person. So she came back on the weekend. I was so locking by saying, that's why boy, you watch your children.
You watch them.
I said, why are you locking them? She said, okay, mommy, I'm locking them. I said, I don't know. Somebody told me that there's something is wrong. This is not my daughter. I always want to chat, tell you stories. I said, not that any problem. Let's solve this problem. So that nobody I will find myself to blame, you know. So after that day when the next morning, I went to social. I went and now I'm not giving the 10 minute problem. She now told me that mommy started crying. I said, I know there's a problem. Cry finish and tell you, it's not that problem. Like I was like, I just want now. You know what I can do? It doesn't happen. Tell me this is what is not my mom will not be going to that end of one. She now said, she listened to this what I said, basically, no problem. She said, no, what is something that I broke my mind? I said, how do you know that? But at least one woman for 5 persons. Hey, I didn't know this.
Wow.
I said, don't worry. It's not life. Let me tell you that. Because before she came back, because those people are telling her that she was, yes. So she left hostel to come back. Yes. So, see, that's why you came back and said, I don't know what to do. So, don't allow them to tell you their leadership. Every end of school, like she has said. Do you understand?
How is she going to get this family?
She just keep on saying.
If you tell your father, even if you can give those on 250.
I said, I used to say that if you have any problem, tell us. Don't worry, don't be afraid. So I said, okay, don't worry, I don't have to tell you that. So the guy came back, they have left in the morning. We wake up and said, you don't be bill. This hijack them, you give them money. I think get the account of the city. So she called for the account. She gave the daddy. So the daddy said, give me the phone numbers of those people. So she now, we were just there. She gave us the numbers that I called. We got 3 of them, and my other people were not available. So we got 3 of them, said, okay, one will give us 3 days. And they paid, I don't want to count. So that was the, I want to see on a Monday because she came back to coming back. That said it was easy, that really to get back. Monday, the other now, they now page 500, 500. Wow. Yes, all of them 500,000. Because it's rich now.
You yourself will see
the hair. So when we pay 2.5, then the last said, okay, that's now that pay 2.5, that's what I'm going to pay. To give us 1 week. So we pay them fully. We pay them. We offset the bill. Because is it that the people disappear?
no, the people
that
they will close the
and they close they will just close the website. No contact. Delete everything. Up to today. So that day we paid it. She was not, she's calmed down and they asked her to go back to the room. And because we started talking with those people, they stopped sending them. So the next week, that was around Thursday, because we paid 500 on Monday. Then Thursday, the other week, we paid one. The father has to look for money and do it. Because it's risky. That's why.
The life is even the life of your child.
That's what I'm saying. Because if you don't do it,
they can do everything.
Yes, and she can even commit suicide before the
I don't know of this betting.
There's one day I heard this morning. There's one I heard this morning. Students were playing gambling in secondary school.
Secondary?
Yes, the teacher went there to stop them.
The
gang of accused
yes,
I heard it.
Oh, your face, yes.
Kill the teacher.
They killed it. They beat the teacher, broke his leg.
When they got to hospital,
the man died. I was shocked.
Yeah, I was wondering that there was nobody else around with that school. You know, they are very well.
And are they on drugs?
They must be. Many of them.
Yeah. I can't remember. I can't just, because children of secondary school are highest 16 years. They're 16 years maximum. Because some of them enter university.
No, this was a government school.
Okay.
And before they came into the school, they
started doing that.
Area boys.
Area boys.
Before they entered the school.
Teacher just walks into his day.
But one thing we really know, that is why it's good to know. We don't know the kind of students you are teaching. That's why we was trying to.
You're supposed to have known.
You're supposed to have known. You don't know your business. Even in France, you can't live their life like that.
You know, I kept on looking at your face.
And you know they are not arrested before.
They're not.
They are not arrested before. They said they went into high school.
They will find each other. Don't like this. Because the parents of those I didn't know your children are asking,
he was a baby.
Blocking the justice to be done.
That's why you should have learned. Because they are trying to no, there are some people they overdoing
they are checking time.
When they are doing
when they are assessing, they check time.
You have the copy.
Don't check time.
You know what I do?
I write the report.
My mama don't carry us.
Okay,
they come back.
Okay.
We're waiting. Okay, good.
When I graduate.
Follow go.
We have to.
You know what I do? Like, if I can't do the exam, the best I can do is to take the paper.
I don't even take papers. I just change their seat. You know, there are some, they are not hesitating.
The giraffe.
When I see, or maybe you are communicating with them, you know, sometimes they say,
no, if I see you with a paper, this paper, I will take it and give it to the exam. The thing is that you are copying and change your seat.
Yeah, I change your seat. If I think you do something like that,
I will just, you see, that's the best thing you can do. The person will not say you are doing a thing to say this and that. Take the script, give the person a fresh seat.
I can go and make another
for our own students, they know that once you have given them, instead of facing panel, they prefer you change the stand for them.
If they feel that they can come to, because they know everything, they do engaging and everything, that will talk with them. And after they have made their if they now come to say because you talk with them, you are now the one defending yourself. Where's the evidence that is talking? And in this case, if they talk with someone, it is not because why are you not coming back?
It's not backfire on you.
Yes, you are the one defending yourself. If I say if I see you, then if you are kidnapping, you just say change your they can come I don't know what to do. No. May not even be able to see themselves right to the other students who saw the paper and the collection
They will write.
They will write that this person goes to paper. Then you and them will now go and face the commission.
Anything can happen. You know my supervisor, I still do supervisor. What happened to do our thing there and I was doing my best. We shot them. Where the two of them did that was shooting a pharmacy shot them. He was killed at him. That is student who failed out. Like after the first second opportunity. To make it. What the dean's law is faculty that is not regulation. Just hired people.
It became his fault.
It became his fault. Hired people. They went in and killed that man. I think after some 2 years,
they will release the person. And even if they do anything, the person has done. Even if they do anything,
the person has died.
The person has died. We are just in what the country is generating. Everything is bad. That's why sometimes I just, when I see people who still defend government, I just wonder.
The person is crazy.
Yeah, they are many in my school. Especially now that they claim that their man is in fact many of them are supporting
because of
right.
You know what in Nigeria is? Bribe and religion.
Yes. If my dad is still there, okay. I should bring him in. No, why I don't want to bring the Professor Jai stand because so that okay. Joseph, keep an eye on our bag for us. My bag and Professor Jai's bag, they are there. Yeah, the next one is my own.
Okay. Joseph, how are you?
It's a bag, see there.
Did you talk to your friend? Okay. I will be sending a message that I met you. I think did you join us in the picture? Okay, let us go and take pictures so that I'll post to him.
Okay.
Now that we are waiting for like so that he will know that you are here with me.
person, the name of the person ought to be in on the first page, like your own. That is one. Two, the petroleum meter that you used, you have to specify the range that you use, because we have 40 to 60, 60 to 80, 80 to 100. Let us know the specific one that you used. And I was just telling my friends here that watermelon, for you to have about 1 kg or 500 grams of the seed to carry out extraction like this, that means you need maybe a truckload. So I just imagine how much you will have bought that compared to the quantity of the oil that you are looking for. So except we are saying, just as Madam suggested here, except we are saying that we already have a particular ailment like cancer, like kidney problem, or anyone that won't use it, you know that you will have substantial effect. That is when one can start advocating that we go into this kind of research. Other than that, it will be like just giving a project to any undergraduate student. Just let him or her do something. Let her go. Thank you very much.
Thank you, our DBC, for that your insightful contribution. So I think, Mr. Abubakar, you have to take note of what. Okay, please, are you there? Can you hear us? Hello, Mr. Abubakar?
Yes, I'm here.
Okay, so you take note of all that our DPC have just, mentioned. And okay, our distinguished mentor here and fellow wants to ask you a question.
Thank you very much. Something on nutraceuticals. Because it's a food plant. So if people can be encouraged to eat it along with the watermelon, that would be fantastic. But we need to take it further. Let us grow the variety, collect the right plant, and so on and so forth. Thank you very much.
Okay, thank you so much, Abubakar. Thank you, our emeritus distinguished fellow, for that insight on people normally eat and remove the seeds. So you can see that it's important that we also eat the seeds so that we get the medical benefits or health benefits. Okay, next question.
The question I want to ask is concerning the purchase of the watermelon from the market. I don't know if you put into consideration the source, because purchasing these samples most times from the market, you are not sure. So did you check the metallic contaminants that are possible to be there in it? Because it's usually more advisable that you collect your sample than buying them. Thank you.
Thank you so much, my daughter, for that question. So, Mr. Abubakar, did you consider the presence of metallic compounds that may be attached through cultivation and all the processes? Did you consider the source of your watermelon? Or did you also check those other contents that may not be very useful to the body, that may be harmful? Are you still there? Hello? Mr. Abubakar, are you still there?
Yeah, ma'am.
Okay.
Can I just make a comment?
Okay, ma'am. Okay.
Just to assist.
Okay, ma'am.
Mr. Abubakar, you are not alone in the journey. Hello, don't be afraid. Even if you are there, don't be afraid. We all culprits.
Yes.
We buy our plant material, even vegetable we eat at home. We don't know whether it is collected from the roadside and brought to the market. And at the end, we be saying we are eating organic, even if we are eating heavy metals. So it's just a wake-up call. The way the country is going now, with the industrialization, this estate, that estate, there were no, all this, Oluku was a dense forest when I was in Benin. But now, 1,000 naira, 500 plot of in Oluku per plot is now multi-million naira. So we need to project to the future. That's what I'm saying. As pharmacognosis, let us start thinking of how to cultivate. But one thing for sure, watermelon, they are not cultivated by the roadside. That one I'm sure of. The other thing about watermelon, if these seeds are really going to be used as nutraceuticals, then we are living good. Most watermelons abroad, they are now seedless. The seedful one, I even hate to eat it now. So, the other thing I know about watermelon, someone is working, doing a fantastic project on the peel of watermelon, that it is highly medicinal. I've forgotten, I don't know the details, but I know somebody is working on it. If really it is medicinal, then all these ones we are throwing away, we know we are in for good things. Yeah. So I think it needs to be encouraged. Mr. Abubakar, we are encouraging you as to pesticides, heavy metals, all of us seated here. Well, thank you.
Thank you, Mama, for this, your insight. Really, we are all culprits to that. And the essence when we are talking about improving yield, trying to benefit or make profits. So that's where we bring all these chemicals to improve yield and make much profit, and invariably also affecting our health. So thank you so much. Let's move on to the next presenter, Mayama Yahaya. You can go on with your presentation.
Good day, everyone. My name is Pharmacognosy from the Faculty of Pharmaceutical Sciences at Nigerian. I'm going to present on the topic potential inhibitors of. Good day, everyone. My name is Mayama Yahaya from the Faculty of Pharmaceutical Sciences, Federal University of Nigerian, Kebbi State. I'm going to present on the topic potential inhibitors of KRAS G12C mutant by ethyl acetate stem bank extract of Vitellaria paradoxa, computational approach. The production of peptide drugs coupled with high cost, side effects, and drug resistance observed in cancer treatment have led to search for alternative cancer drugs through screening of medicinal plants. And in this research, Vitellaria paradoxa has been utilized. Now, 9 grams of ethyl acetate extract have been used for the purpose of chromatographic studies. And we have used silica gel as stationary phase, while the combination of N-hexane and ethyl acetate have been used as the mobile phase for the column chromatography, where a total of 342 collections have been made, which were put together based on their TLC profiles into 7 major collections. And out of these 7, 3 of them have given a single spot on TLC, and also gave some unique colors, the white crystals, the brownish color, and pink substances. For this purpose, we have selected these fractions for characterization, and we have coded them ME311, ME323, and ME332. The characterization of these compounds have been done with the aid of LCMS, NMR, both 1D and 2D, and also finally with comparison with literature. And the compounds characterized have been subjected to in silico studies to conduct molecular docking and ADMET studies. ADMET as in absorption, distribution, metabolism, excretion, and toxicity. Now, the 3 compounds have been docked against KRAS G12C mutants using the software or the online server called Doctor, while the post-docking analysis have been done with the aid of Discovery Studio. And finally, we have used Sweets ADME and Protox 3 for the ADMET profiling. The LCMS shows the molecular formula of the 3 compounds and also the molecular ion peak values for the 3 compounds. This is the comparison of the spectral data of the 3 compounds with literature. This is the proposed structure of the first compound, ME311, and the name is catechin. We have the second compound as catechin 3-organic, the third compound, gallocatechin, and all these 3 compounds belong to the flavonoid group. For the docking scores, catechin with minus 8.481 has shown the highest binding affinity with the protein. allow actions of the three compounds, including the protein. Highlighted in red are glutamine and thyroxine, two of the amino acid residues targeted by anti-cancer drugs. And we can see that all the three compounds are bound with these amino acid residues. In terms of solubility and drug likeness, the three compounds have shown favorable ADMET profiles. In acute oral toxicity class of 6, 4, and 6, and also predicted LD50 of 1000 and above, the three compounds are considered safe for oral consumption. So, with the results obtained in this research, we can conclude that these three compounds are potential sources of anti-cancer drugs, especially for lung cancers associated with tobacco smoking. Thank you.
Thank you so much, Yama, for your presentation. We moving fast. I'll entertain one question from the audience. Okay.
Hello. Hello, can you hear me? Okay. All right, can you hear me, please? Hello?
Yes, I can hear you now. Go ahead.
Okay, I wanted to ask, thank you very much for a very beautiful presentation. Now, I was looking at your affinity binding scores, and I was wondering that you said one of the compound was -8 point, the highest compound there. What was that again, please? You can remind me. -8. So I wanted to ask, your root mean square deviation, how was it like for that compound?
I'm sorry, sir. Are you talking about the dot scores?
Yeah, the docking scores, yes. The first compound.
Okay.
Yes. What was your root mean square deviation for that compound, comparatively to the standard, if you have the standard at all? I didn't see the standard in your docking scores.
Okay, sir. I actually have not done any of the root mean square calculations, but these scores are given directly by the doctor server, and it is automatically generated after the docking. And the fact that this -8.481 value is the most highly negative value, that means it is giving us the highest binding affinity, because the lower the affinity, the higher the affinity. That's the way it happens.
Please, that's not good enough. What of the standard? Do you have the standard? You have the score for the standard. What did you use as the standard?
The last group you can see sotorasib is an anti-cancer drug.
Let's go to the slide. Let's see.
And
slide, I think that's towards the end. Move backwards. No, binding score. Go to the back. Next, backwards. The table where you have 8.41 something, 7 point minus scores. No, go forward. Move forward. Move forward.
It's not done yet. It's not changing.
Yes, here. Is the last one the standard is the last one. I'm just seeing it.
Okay.
Yes, that's the standard. We can see it now. -7. So that one is higher than it actually. Okay. So take the lower. Yeah. So thank you so much for your presentation, and we now move to the next presenter. Thank you.
Thank you. Thank you very much. Thank you.
Good day, distinguished professors, colleagues, and fellow participants. I am Salifina Hawal Aboita, and I'll be presenting on the study Pharmacognostic Evaluation of Herbal Medicine containing leaves of Osimo santonin used in the management of COVID-19 symptoms. This study was carried out in the year 2021 and was supervised by Professor Hajara Ibrahim of the Department of Pharmacognosy and Drug Development, Ahmadu Bello University, Zaria. COVID-19 pandemic results from viral infection caused by SARS-CoV-2, and since viral infections oftentimes are self-limiting, treatment has been focused on symptoms management. The pandemic led to a global reliance on natural remedies for symptomatic relief. On that note, plants like Osmond santon, ginger, clove, and turmeric were used alone or in combination for symptomatic relief. Due to the presence of bioactive compounds with immune-boosting, anti-inflammatory, antiviral, and antitussive properties in those plants. Hence, there is need for scientific evaluation and standardization of these herbal formulations to prevent adulteration, improve safety and efficacy. The aim and objective. The aim of the study is to evaluate and standardize the traditional herbal remedy containing leaves of Osimo santon used in the management of COVID-19 symptoms. The objective is to perform microscopical and phytochemical evaluations, quantify total flavonoid and quercetin, conduct elemental zinc analysis, and also compare extraction yield with and without tea bags. Materials and method. The plant material listed below were collected and identified at the herbarium unit of the Department of Botany, Ahmadu Bello University, Zaria, and the herbal formulation was prepared using the formula 1/2 teaspoons of Osimo santon, ginger, turmeric, and 2 pieces of clove, giving us an average weight of 4.4 grams. Extraction yield was water extraction with and without tea bag. The pharmacognostic analysis gave us microscopical evaluation, phytochemical screening using the standard tests, HPLC to quantify quercetin, atomic absorption spectroscopy for zinc. Then the result we obtained was we detected compounds like alkaloids, saponin, flavonoids, tannin, and other bioactive compounds with known antiviral, antioxidant, and immune-boosting effects. The extraction yield, we got better extraction yield from the tea bag. Reason being that the pore size of the tea bags were way larger than the filter paper that was used for extraction without tea bag. The quantitative analysis, we got the total flavonoid to be 114 milligram per gram, the quercetin to be 0.00333 milligram per gram, and the zinc to be 0.136 milligram per gram. For the discussion, the results of this study are in line with previous studies that reported that Osimo santon contains certain bioactive compounds that are responsible for its antioxidant, anti-inflammatory, and antiviral properties. It was also reported that 3.5% flavonoid yield was gotten from ethanolic extract of Osimo santon. However, our study gave 11.4% flavonoid yield, which may have resulted from the combined contribution of flavonoids from other components of this herbal mixture. Zinc gotten from this study also support earlier findings on zinc's immune modulating and antiviral role, as well as quercetin, which is characterized with antioxidant, anti-inflammatory, and immune modulatory properties. The combination of all these three properties makes quercetin as well as this herbal formulation as an excellent candidate for dealing with situations in which oxidative stress, inflammation, and immune systems are involved, just like in COVID-19. Therefore, this result confirm the pharmacological relevance of the formulation and justify its traditional use. The conclusion is that this study has established the pharmacognostic and phytochemical standards for the herbal medicine which contain key bioactive constituents with proven therapeutic relevance in managing COVID-19 symptoms. Hence, the standardization of such herbal medicine is essential to ensure quality, safety, and efficacy. The findings from these studies therefore provide baseline data that can be used for quality control and standardization of osmol santon-based formulation with the presence of quercetin and zinc serving as a reliable markers for standardization. Recommendation. Further clinical and toxicological studies are recommended for long-time safety validation. Also, incorporating tea bags add value for herbal medicine, and an infusion time of 5 minutes or more is recommended for optimal yield. And here are my references. Thank you for listening.
A round of applause for her. Thank you. Hello? Hello?
Yes.
Okay. Thank you for your presentation. Please, we welcome questions from the audience. One question from the audience.
Okay. If you are hearing me, what's the difference? What's the difference between your plant, Osimo santon, and Osimo gratissimo?
Did you hear?
Okay.
Yeah.
Yes, I got the question. Okay, the Osimum santon, it has, the leaves are smaller than the leaves of the Osimum gratissinum, and usually from the microscopic features. And usually the leaves of the Osimum gratissinum is usually raw, while the leaves of these Osimum santons are smaller and smoother.
Yes, that shows you are part of that work. Thank you so much for the answer. Quite correct.
All right.
So thank you for your presentation. Next presenter, please.
The chairperson and our distinguished audience, good day all. I am by name Talba Saleh Hamawa, a postgraduate student from the Department of Pharmacognosy and Drug Development, Ahmadu Bello University, Zaria. On behalf of my co-authors, I am here to present the finding of our research titled Pharmacognostic Standardization and Antibacterial Evaluation of the Leaves of Borrelia radiata. Antibacterial resistance has become a global challenge and is among the top 10 threats to a global health. This global challenge underscores the need to explore plant-based therapeutics, of which Borrelia radiata is a plant from a Rubiaceae family, which has been traditionally used in various herbal formulations for the treatment of bacterial and viral diseases, but lack adequate scientific characterization and antibacterial evaluation. This study was aimed to establish a microscopic physicochemical standards, carry out the phytochemical screening, and evaluate the antibacterial activity of the aqueous leaf extract of Borrelia radiata against clinical pathogens. The cytomorphological and the physicochemical studies of the Borrelia radiata leaves was conducted according to established standards. Then the phytochemical screening too were conducted, and the antibacterial screening. Here are some of the results of our findings. The first image there is the plant in its natural habitat, that's the Borrelia radiata, while the second one under it, that's the B, is a photomicrograph of the abaxial epidermal layer of the Borrelia radiata. On the picture, you see that there are two features that were identified, which are an homocytic stomata and a wavy wall epidermal cells. Then on the third image here is a photomicrograph of the adaxial epidermal layer of the Borrelia radiata, where a cicatrix and a polygonal striped wall epidermal cells were identified. Then on the fourth one is a transverse section of the leaf of Borrelia radiata. This shows that the leaf is a dorsoventral leaf That means it has like a distinct adaxial and abaxial surfaces with a different diagnostic features. Here we can identify like a palisade mesophil, a spongy mesophil, a vascular bundle, serrate covering trichomes on the surface of the picture. These are the quantitative and the physicochemical parameters of our findings. Table 1 is the quantitative microscopical values, where all the values that are being seen on this table are the average value of 5 counts and the standard error of a mean. While the table 2 are the physicochemical constants, such as the moisture content, total acid insoluble, water soluble, and the extractive values. All these ones are the average value of 3 determinations and the standard error of M. These are the results of our physicochemical screening, of which almost all the plant constituents that were tested happens to be present with X. It's only a mixed reagent, that's alkaloid, that didn't react or that give a negative result. But the trigental and the Wagner reagents reacted, which signify the presence of an alkaloid. So that maybe the alkaloid is in trace quantity. That's why it couldn't react with a mixed reagent. These are the results of our antibacterial studies. Table 4 is on of inhibition, which is a millimeter at various concentrations, that's from 100 to 12.5. And ciprofloxacin, which is a standard control drug at 10 microgram per ml, which gives all these values. That's the zones that it was able to inhibit the growth of the organisms. While the table 5 is the minimum inhibitory concentration. This is the minimum concentration of the extract at which no any sign of a growth was observed. But under this condition, we cannot be able to justify whether the concentration killed the organisms or it just inhibits the growth. That's whether it is bacteriostatic or bactericidal. That's the sense that leads to the determination of the minimum bactericidal concentration, that's the MBC. That's the concentration at which the extract kills the organism, of which most of them, or all the organisms, were bactericidal at 12.5 milligram per ml concentration. While from the previous table, you'll identify that some of the bacteria were inhibited at 6.25 milligram per ml, while others were inhibited at 12.5 milligram per ml. In conclusion, Border radiata showed distinct microscopic and physicochemical standards with rich phytochemicals. Its ethanol extract exhibited a strong antibacterial activity, validating its traditional use and potential as a natural antibacterial or antimicrobial agent. These are some of my references.
A round of applause for Hamawa. Thank you so much for your presentation. Questions? Okay? You have to come close to this place.
Thank you very much for your presentation. The slides are wonderful. Your microscopic slides are wonderful. But I want to ask, did you carry out tests for calcium oxalate, difference of calcium oxalate on the plant?
Okay, Mr. Hamawa, did you get the question?
Yes, ma'am, I got it.
Okay, they said, did you study the presence for calcium oxalate? That's chemical microscopy. Did you carry out that study?
Yes, I studied. Yes, I've carried out that one. The calcium oxalate was present on the surface preparation.
How did you confirm the presence?
Yeah, and after dropping a concentrated HCl, so the duress type of calcium oxalate were all over the surface of the plant, surface of the on the surface preparation of the plant.
If you add HCl, what will happen to the calcium oxalate?
They dissolved.
Fine. Okay, thank you so much. So I think that's all for H2SO4, that's the mistake, HCl. So, the person asking is saying you use HCl instead of H2SO4. So, but that's the way.
Yes, ma'am.
Okay, I was just trying to say that you're supposed to use H2SO4 instead of HCl, called sulfuric acid.
All right.
Okay, that's noted, but you observe the disappearance. So we move on to the next presenter. Thank you so much for your presentation. Next.
Thank you.
How many left? 3. Okay. Let's move fast.
Good day, my President, highly respected and distinguished professors, and good day to everyone. I am Adeniran Adeyako. I'm presenting on cytotoxic and growth inhibitory effects of Garcinia colastomac, a potential bioactive inhibitor against HDAC8 using molecular docking. I'll be guided by this outline, and here is a graphical summary of my presentation. Garcinia cola belongs to the family Cruciaceae. Is a valuable medicinal plant in West and Central Africa, and traditionally it has been reported in the treatment of malignant tumors. Recently, overexpression of histone deacetylases, HDACs, has been attributed to tumorigenesis in several cancer types like liver, lung, cervical, colorectal, just to mention a few. And they actually play a progressive role in cancer, and because of that, they are prime targets when it comes to cancer therapy. Figure 1 shows the stem bank of Garcinia cola collected from Otopani local government area in Cross River State. The objectives of the study is to evaluate antioxidant, cytotoxic, and inhibitory activities from the extracts and fractions of Garcinia cola, and also to evaluate the potential use of the identified compounds from the N-hexane fraction against histone deacetylases. Materials and methods. Pulverized powder of the stem bark, 500 grams, was extracted in methanol and subsequently partitioned in N-hexane, dichloromethane, ethyl acetate. And of course, the aqueous fraction was also kept. These fractions, alongside with the extracts, were subjected to in vitro antioxidant activity using DPPH, cytotoxic activity using branched and growth inhibitory assays using sorghum bicolor-6. The N-hexane fraction was subjected to GCMS analysis, and of course the identified compounds were also molecularly docked, and their molecular dynamic simulation were also obtained. Results. The inhibitory concentration of the free radical scavenger activity was highest in ethyl acetate fraction with 16.7, followed by N-hexane with 25.3, as compared to ascorbic acid having 12.2. For the leader concentration of the cytotoxic study, the N-hexane and the DCM fraction had 5.5 and 5.0 microgram per mil, as compared to the standard cyclophosphamide, which had 58.0, as shown in figure 2. Similarly, the N-enzyme fraction was most potent in the inhibitory effects using sorghum bipolar. At 3 mg per ml, there was a reduction of 76% in the length of the radicle of the seed as compared to the control, as presented in figure 3. Figure 4 shows the GCMS chromatogram of N-enzyme. Table 1 also shows the molecular dot compound from the GCMS hits with the HDACH protein. And we observe that quercetin 3O galactoside had the highest binding affinity of -9.5A3 as compared to other compounds. Figure 5 shows the 2D interaction profile of quercetin 3O galactoside, and of course the amino residual bondage to hydrogen atom there. In conclusion, our results suggest that N-enzyme function from the Garcinia cola of methanol stem bark extract possesses potential cytotoxicity to the nuclei of shrimp, as well as inhibiting the growth in the length of the radicle of sorghum bipolar seeds. The molecular docking results also reveal that quercetin 3O galactoside demonstrated the strongest affinity for the HDACH protein, and it also forms a stable interactions with the binding sites and establishing 3 prominent hydrogen bonds with amino acid residues. So these results suggest that quercetin 3O galactoside could be potential bioactive inhibitor from the N-enzyme fraction of Garcinia cola, possessing antioxidant, cytotoxic, and inhibitory activities. These are some of the selected references. Thank you for listening.
A round of applause for Adiniran. Thank you so much for your presentation. Questions from the audience? Okay.
All right. Thank you very much for your presentation. I wanted to ask a question.
Thank you, sir.
Yes. On your last slide, when you talk about quercetin 3O galactoside, right? You said this is yes. Now that compound, as it were, do you expect to find that in adhesive fraction of your extract, because quercetin itself is flavonoids that are rarely found in adhesive fraction. Not only that, it comes with a galactoside. So you have very highly polar compound here. How could you have found that in the adhesive fraction?
Yes, thank you so much. It was actually surprising, like you said, because the quercetin and with the galactoside attached to it, which actually makes it a soluble compound. But from the GCMS analysis, from the result we got, we actually found out quercetin 3O galactoside. And preliminary, when we phytochemical screening from the extract actually also suggests, that it contains flavonoids. But like you rightly raised, it's somehow somewhat actually surprising for us, but that was actually what we got from the GCMS analysis.
Do you know the problem, where the problem is coming from? Can you guess?
Yes, ma'am.
So tell us where you think. Good.
Hello? Yes, ma'am.
Yeah, that shows improper fractionation. When you were removing the non-polar from the polar, because the first fractionation, you find out that polar, N-hexane will be on top. and the heavier one will be below, which is the. So when you are running off, you're supposed to run off the heavy one, which is below, and then run off the hexane. So the likelihood that you didn't totally remove the aqueous methanol extract, for you to now remove your N-hexane on top. So that's fractionation error. So you still have little of that in your N-hexane before sending it for GSMS analysis. So that's where it's coming from. Did you get that?
Yes, thank you so much, man. Even though we use TLC, to monitor during the partition, but like you already said, it's possible that the extraction was not complete.
All right.
The partition was not complete.
I would suggest to you, sir, are you hearing me?
Yes, sir, I'm with you.
Using GCMS in this case might not do justice to this work. So I will expect that you go ahead and carry out HPLC analysis. That should be able to identify, especially when you use standards, better standards like Pastine and the rest of them, and you give you a better result to actually find out that this compound is not in the hexane fraction.
All right, sir. It's noted. And the effort is also going towards that direction. In fact, in isolating and characterizing.
Yes, so you have to fractionate it again. Get the extract and do the proper thing and eliminate all the errors. Then you get where it belongs. Okay. So, another question. Let's be fast.
Thank you very much, Mr. Eniro. I'm only interested in how you carry out your cytotoxicity, and perhaps you share the results, because it's there on the title, but I'm not really sure whether you display how you carry out your cytotoxicity and the results. Thank you.
For the cytotoxicity, we actually use a blank shrimp, and we hatch it using seawater, and then we prepare different concentrations ranging from 1,000 microgram per mil to 1 microgram per mil, and then following the hatching of the brine shrimp after 36 to 48 hours, 10 of the nopla will actually move, into our vials in replicates. So at the end of 24 hours, we were able to count, the number of active, in terms of their mobility, whether they are still alive or dead. So that's how we arrived at the legal concentration result we are seeing.
Okay, thank you so much for the presentation.
Thank you, ma'am.
You're welcome. So the next presenter.
Every day we enjoy the taste of the ocean fish. It sustains us, defines our cuisines, and connects us to our roots. But many people are unaware of the composition of this proteinous food. And this brought us to the topic physicochemical profiling, total flavonoid and total phenol content of the oil extract of Pangasius hypothalamus fish, Pangasidae. I am Isimot Arono from the Department of Pharmacognosy, Olabisi Onabanjo University. Fish oils are well recognized for their nutritional and therapeutic properties, owing to their richness in essential fatty acids, antioxidants, and bioactive compounds. Pangasius hypothalamus is a marine edible fish, locally called Eja Obokun, and it's widely known for its oily nature and dietary value. However, the physicochemical properties of its oil constituents remain underexplored, particularly in Nigeria. Hence, this study aims to evaluate the physicochemical characteristics of Pangasius hypothalamus oil, to quantify its flavonoid and phenol content as well as chemical constituents of the oil. Method. The fish was obtained from a cold room at Abule Egba in Lagos State. It was carefully transported under ice to the laboratory. Thereafter, it was thoroughly rinsed and deboned. The flesh dried at 60 degrees Celsius for 3 days. Thereafter, the dried flesh was then blended and macerated using N-hexane for 72 hours. The filtrate was filtered and concentrated in vacuum to obtain the oil extract. The oil extract obtained was then subjected to physicochemical analysis, including acid value, saponification value, using the titration method. The quantitative analysis was carried out, including total flavonoid content using the aluminum chloride colorimetric assay, total phenol content using the Folies-Yokatu assay, and vitamin E content was also quantified. GCMS analysis was also carried out. Results. Table 1 shows the physicochemical analysis of the oil sample, and we found out that the acid value of the oil sample was 4.91 milligram of potassium hydroxide per gram of the oil. Saponification value was 166.4 milligram potassium hydroxide per gram of the oil, and we observed that the oil melts at 37.5 degrees Celsius. Table 2 shows the quantitative analysis results. The amount of flavonoid content in the oil was found to be 194 microgram of routine equivalent per gram of the oil sample. Phenol was found to be 40 milligram gallic acid equivalent per gram, and vitamin E was found to be 18.3 milligram of tocopherol per gram of the oil sample. The GCMS analysis in Table 3 revealed or identified the presence of carotenoid, fatty acids, and biogenic amines. Discussion. Carotenoids are known for the antioxidant protection. They help in supporting the immune system and eye health. Fatty acids are known for the anti-inflammatory property. They help in hormone production and also serve as emulsifiers in pharmaceutical industry. However, we observe slightly elevated amounts of biogenic amines, which might be as a result of handling, post-harvest handling, and storage. We conclude that Pangasius hypothalamus oil possesses nutritional quality, demonstrates antioxidant and nutritional benefit through the flavonoid and phenol content, and this study provides a baseline data for future fish oil research in Nigeria. We recommend that there should be an improvement in post-harvest handling and storage of this fish. We also recommend that further study be conducted on the oxidative stability and safety of the fish oil, and the findings should be integrated into nutrition and aquaculture program. We recommend and encourage that there should be a local industrial cultivation and oil production of this fish. These are some of our selected references. Thank you for listening.
A round of applause for Arowana. Thank you so much for your presentation. Questions? Okay.
Thank you, ma'am.
You're welcome.
Thank you very much for your presentation. Well, I was just thinking that oil will have been characterized alongside with a standard oil, so that to be able to really appreciate its value and compare its effects with the existing one.
All right, noted.
Okay.
How did you get
No, he studied total flavonoids.
Thank you very much, Dr. Rona. Thanks for the work. It's good work on animal natural source, as it were. I was just wondering, did you confirm this flavonoids present in fish before you went into it? Did you confirm?
Hello?
Did you confirm the presence of flavonoids ab initio before you went on to quantify them? And are flavonoids found in fish? Are they not plant products? I'm just wondering. I want your answer.
Don't wondering. It's not only you.
Thank you, sir, for the question.
Okay, the question is, okay, you are ready. Continue.
Hello, ma'am.
You can continue.
All right, thank you, ma'am. Yes, ma'am. The reason why we check the flavonoid content of fish is because we are, as we know, fish is one of marine organisms that are available, and this fish, we observe that apart from the fatty acid that we know that is present in the oil, we also observe, because of large amounts of volume of oil that the fish contains, and that was why we just look at checking the flavonoid level of that is present in the fish oil.
What we are saying is, if you can hear him right and the views of others, you can't be studying total phenolic content of an oil, right? So that's okay.
Yeah. I think what we are trying to contend here is that you listed that you found flavonoids in the fish. How did you get there? That's the question. You know?
Yes.
Now, if you carry out anti-oxidant activity, I can also tell you that the vitamin
Sir, if I get the question, sir,
did you really find flavonoids in the oil, or you just imagine that? Yeah, that's the question. Did you hear me?
No, we quantify.
Yes, you quantify. How did you get it to quantify? And that's the point.
What's the rationale?
Did you get the question, please? Hello, are you there? Hello, are you there?
Let's go to yes. So I think the take home here is that it's not obtainable, you know, and maybe you need to go back and look at the methodology and the title if you really what you wanted to study. You go back and look at, okay. Comments, just hold on.
Well, much as we agree with what everyone has said, of course I feel that this kind of work ought to have been, GTMS ought to have been used. That would have been better than this one. Because the equipment, I was just telling Prof, when they talk of phenolic, total phenols, total flavonoids, the equipment that Oyiboman made for us, sometimes you see the flavonol content of hexane fraction more than that of the methanol fraction. And people will report it, and that is not rational. It's not something you can see at all. The same way we use HPLC. Some HPLCs have just been laced with certain library data, artifacts. Like once you just put your own, it will show Kenferol, it will show Kenferin, it will show this one, Constantine, everything. You bring another plant from anywhere, it will show the same thing. So there is need for us to be circumspect and also know what the kind of analysis that we are doing. Thank you, ma'am.
Okay, yes, thank you, our DBC, for the insight. You know, sometimes I quarrel with my students. When you bring data, I will tell you it's not true. Because there are bases. There are things that are obtainable. It's just like when somebody tells you an alkaloid found present in hexane. So you begin to imagine, dream, or what. So sometimes we need to have knowledge of these basics so that even when you get your results, because what our DPC just said is, it might be what happened to your work. We are not there, but sometimes we need to know things that are possible and the things that are not possible. We still thank you so much for your presentation. You take home to take note of these comments and see how you can look at the work, for further studies. Thank you so much, Arowona, for your presentation. Next presenter.
Objectives of study. Sickle cell disease is one of the studies that are derived by an influence of an medicinal treatment. Sickle cell disease.
Hold on. Who is presenting? Hold on. Hold on, please. Go back.
Objectives of the study. Sickle cell disease is one of the studies that are derived by an influence of an medicinal treatment. Sickle cell cells tend to block capillary chamber. Objectives of the study. Sickle cell disease is one of the studies that are derived by an influence of an medicinal treatment. Sickle cell cells tend to block capillary chamber. The plant most nutrient, the mode of preparation and administration of plant is one of the most common life-threatening and also to determine the some indigenous plant and plant families with high methodology. The survey was conducted in June-July 2025 in 11 local governments, covering all the 3 senatorial zones in Kenya. The information were obtained through semi-structured questionnaires and distributed to the respondents. The respondents include herbalists, herbal medicine sellers, farmers, teachers, elders, and others. A notebook and a pen were used to document the local names of the plant. A camera was used to take pictures of the medicinal plant. Other data collected were parts of the plant, method of administration, and method of preparation. Descriptive statistic and ethnobotanical index was used in the study. Results and discussion, demographic features. A total number of 105 respondents participated in the survey, and most of them, that's about 56%, were above 50 years of age, 59% were herbalists and herb sellers, 81% of the respondents were male, while 19% were female. 45% have more than 20 years experience, and 80% acquired their knowledge through oral tradition. This is the table for the demographic information authority. 99 plant species belonging to 45 families were discovered in the survey. The plants with the highest frequency are Ficus platyphylla, Boswellia odorata, Laptedemia hastata, and Nigella sativa. And this thought the family Fabaceae has the highest number of 23 species, which include Tamarindus indica, Laptedemia hastata, Cassia occidentalis, Deuterium microcarpon, followed by family Poaceae, Rubiaceae, Combritaceae. This is in line with earlier reports of Amuye Bay et al. 2016, Badamasi et al. 2020, Cyril Omotayo 2020, who indicated that Euphobiaceae and Fabaceae were the most prominent families used in the management of sickle cell disease by traditional practitioners in Nigeria. The stem bark of the leaves were found to be the most used part of the plant. Cyril Omotayo also reported a similar finding. Decoction and oral administration were the commonest methods for preparing and administering. This is the table showing the ethnobotanical survey and some of the ethnobotanical survey. Summary and conclusion. The ethnobotanical survey reviewed in this study successfully identified 99 medicinal plants from 45 families that are used by the traditional practitioners in Kenya. Higher percentage of the family came from the Fabaceae, Poaceae, Rubiaceae, and Combritaceae. The plant with the highest frequency are Ficus platyphylla, Boswellia odorata, Laptedemia hastata, Nigella sativa. The stem bark and leaves were found to be most
of the plant used. Decoction and oral administration were the most commonest method of preparing and administering the plant. These are part of friends. Thank you for listening and have a nice day.
Thank you so much, Abdul Aziz, for your presentation.
Thank you, ma'am.
Questions? Okay.
You mentioned the collected some plants. The ones you collected, did you get the specimen number or the addendum number? One. Then two, for those ones that has a kind of strategic information, like the most frequently mentioned or the family mentioned, at least a picture of them would go a long way to help those of us that are interested in such. Thank you.
Hello, Abdul.
I didn't get your first question, but the second question, if I get you, you were talking about the display of the pictures. The reason I had a lot of slides while I was having challenges with the time and the number was limited. So I had to continue reducing the slides. So it made me to reduce a lot of slides that are of importance. So that is why I was not able to display the pictures, because it is somehow heavy and we have a limited load of what to upload.
What of your voucher number?
What did you say, ma'am?
The voucher number. She asked for voucher number.
Yeah, voucher number. Yeah, sorry, I took the plant to NDA for identification, but unfortunately they did not release the identification number until this morning. In fact, if there is a way I can share it to you, I would have shared it. I have my complete voucher number, 99 of them. It's there. I just received it this morning.
That's interesting. That's interesting that you're able to have a voucher number for 99 plants. That means you collected all of them. That's interesting. For the picture.
Yeah, I collected the plants. In fact, I even pressed the plant. I have the pressing. I pressed the plant. I collected the individual plants. I pressed them, and I took them to the herbarium of NDA. The plants were identified, and the voucher number was issued. It's just that it was delayed, but it was released this morning. I have them with me now.
Thank you. That's interesting. That's what I said. That's what I said.
Okay, thank you very much for that comment.
For the picture,
thank you very much.
One of your slides, just pick a critical, strategic ones. You don't need to read the slides. You just click through it, but we have it. People have questions to ask already. And then another thing, another observation, because I've done a lot with botanical studies. At times, plants may have different local names. Did you come across such? Because when you say you have a 99 voucher specimen, did you hear me? Hello?
Hello? Hello? Abdul, are you still there? Okay, I think we have. Okay, thank you so much. The last presenter, please, so that we go over to the physical presentation. This has been presented, please.
Heritage Chairperson, distinguished colleagues. I am Sadio Pedro Abdulaziz from the Department of Pharmacognosy and Drug Development, Ahmed Bello University, Zaire. I'm here to present our work titled Pharmacognostic Evaluation of Medica Foetida, LEAF. Herbal medicine remain vital to global healthcare, particularly in developing countries where about 80% of the population depends on them, according to World Health Organization. Despite the extensive conventional use of Medica foetida in treating fever, cough, stomachache, and malaria, there is limited pharmacognostic data on the plant. This gap can lead to adulteration and misidentification, which compromise quality and safety. Our objective is to establish the comprehensive pharmacognostic standard for the leaf of Medica foetida, encompassing the microscopic, microscopic, and physicochemical parameters. The fresh leaf, as you can see here, the fresh leaf is broadly ovate. It is heart-shaped with a deeply coded base and serrated margins. When crushed, the leaves emit a very strong offensive odor, and microscopically, the adexia and the abaxial layer has two different types of epidermal cells. And the adexia layer will have the wavy type of epidermal walls, while at the abaxial layer we have the straight polygonal wall, and at the adexia layer we have the anomocytic type of stomata, while at the abaxial layer we have the anisocytic type of stomata. Then the triangular section of the middle reveals the multicellular uniserrates covering trichome, and we have the vascular bundles around here, while the upper epidermal layer is around here also, and the lower epidermal layer can be found around here at the triangular section of the leaf. Then these two tables show the microscopic profile of Medica foetida leaf. The first table shows the quantitative features of Medica foetida leaf. The quantitative feature shows the consistent values across vein island, vein termination, stomata number, stomata index, and these parameters help distinguish Medica foetida from closely related species. While the other table, the table 2, shows the micrometry of Medica foetida. The micrometry evaluation further characterizes the dimensions of the epidermal cells, the stomata, and establishing measurable diagnostic data for reference purpose. Here we have the chemomicroscopic and physicochemical profile of Medica foetida leaf. In the chemomicroscopic analysis, it revealed the presence of key cell wall materials like the cellulose, lignin, suberin, and 15. They were all present, and also some key cellular inclusions such as calcium oxalate crystals, starch grains, and the rest. These features provide additional authentication markers and contribute to understanding the plant's structural composition. Also, the physicochemical constants, including moisture content, total ash, acid insoluble, extractive values, were determined to establish the purity and quality profile of the fruit drug. All the results fall within the acceptable limits according to World Health Organization for fruit plant material, including indicating minimal contamination and good storage facility. These parameters are vital in assessing sample integrity, detecting adulteration, and standardizing herbal formulation derived from Medica foetida. In conclusion, the diagnostic pharmacognostic parameter established in this study provides essential baseline data for identifying and authenticating Medica foetida leaf. They form the foundation for its quality control and standardization as a food drug. Overall, our findings validate its traditional use and support further phytochemical and pharmacological studies aimed at developing standardized herbal preparation for these plants. Thank you for listening.
A round of applause for Saliu K. Thank you so much for your presentation. Question from the audience? Do you have any? Okay.
Thank you very much for your presentation. During your presentation, you made mention of some other species of the Medica. Please, can you mention other species that that this Pharmacognosy can be differentiated from, that are readily available?
Thank you for the question.
Hello, did you get the question? Hello, Salu. Did you get the question? Did you get the question? Okay.
I said, during the course of the presentation, the mention of certain characteristics that this Pharmacognosy species exhibited that differentiated it from other species of Momodica. I was now asking, can you tell us other species of Momodica that are different from this Pharmacognosy?
Okay, thank you so much. Any other question? Okay, in absence of any other question, we have come to the end of the visual presentation. I want to thank all of us for time spent. I want to thank us for our wonderful contributions from our elders here. I want to also thank the assessors with me. Professor Binadoa and Professor Mrs. Organ. She has left to attend to some persons waiting for her. So I thank all of us and pray that we continue to the next presentation, which is going to be the physical presentation. And I'm happy we are all learning from this process. Thank you, and thank you all.
Good afternoon, everyone. Standing on the existing protocol, Tunde Holabi, a PhD student of Pharmacognosy department in this great university, under the supervision of erudite Professor B.A. Ayinde. I'm standing before you this afternoon to share some of our findings from our ongoing research titled Bioactivity Guided Isolation and Characterization of Anti-Cancer Constituents from Praxelis clematilis. By the way of introduction, or rather before I go on, I want to believe that in this hall and virtually we are convinced that plants, medicinal plants, have a lot of potential. If we still have one or two persons that is having doubts, I just have a statement for such an individual. And the statement, I hope my supervisor will permit me to use his statement, which goes, a trial will convince you. So such an individual should perhaps bite, let's say, Plumbago xylonica, the root, or if that is far from fetch, if that is far from fetch, you can slice and soak datura methyl fruit and sip that after a while. So perhaps after consciousness, such an individual will live to testify to the potential of medicinal plants. Praxelis is one of such medicinal plants that harbor such potential, and it is not known for its medicinal importance. Rather, it is known as an invasive plant, so as much that an environmental organization in the United States of America has classified it as a plant of particular concern. And you remember the President of the United States, of course, recently said America, they are very fast and swift when it comes to decision making. So this is the plant, and of course, another very key important thing about this plant is the striking similarity between this plant and Agerantum conizoides. And it belongs to a family, Asteraceae, and we all know that Asteraceae member, they play important roles in oncology. So that's part of the what formed this research. Another thing is that we know that orthodox drug has made advance and enormous contribution when it comes to the treatment of cancer. But of course, there are a lot of side effects. For example, the life-threatening side effects of these anti-cancer drugs, such as nausea, vomiting, dysentery, and what have you. And the second thing, cost of treatment, of course, is very expensive. As of today, the average cost of cancer treatment is about $100,000 US, which is equivalent to 100 million naira as for today. And another thing is availability of facility, of course, as Professor Emeritus said. Even when these facilities are available, they are very, very inaccessible. So, as for what we have done and how we have done the things that we have done, the chemical use are normal chemical, of course, including Cephades L20, and the plant collection and identification. After the identification of the plant, the first thing was to cultivate the plant. Of course, my supervisor told me we might run into problems during dry season. So, I was very lucky. Is just sprinkle the seed and it began to grow, and it even became problem to eradicate this plant. So afterwards, the plant was prepared and extracted using succinate extraction. Methanol extract was obtained and biological activity were done. The biological activity
2 minutes more.
Yes, the biological activity done on the plants include growth inhibitory, lysosomal bicolor, and cytotoxicity using tadpole. The extract was partitioned into two extreme polar and non-polar solvents, DCM and water, and VLC was carried out on the most active fraction. Then column chromatography was done, of course, monitoring the process with TLC. Along the line, preparatory TLC was equally done, and further purification was done using Cephades L20. As for the results, you can see the growth inhibitory results now. The plate A is for the control. When compare the control with the crude extract, you see the growth of the radical for the Genecon is so obvious for the control. Then the slide C, D, and E. D is the control, while C is for that of Achios, and E is for the DCM fraction, and we obviously see the reduction of the goods. And of course, the graph there we have at 10 milligram, you see the DCM fraction is having a very reduced growth. Then for the toxicity, we have at by 2 hours, we already have 100% mortality for the DCM fraction. And as for the TLC plate, of course, this first one is the VLC fractions and how we were able to combine these. then the last one, that wider one, is for the preparatory TLC. And of course, this, after the process of isolation, we're able to isolate about 9 compounds. And of course, to be sure that this plant is actually, it contains cancer, anti-cancer constituents, we have sent this isolate and the fraction for biological screening on cancer cell line abroad. So these are the ongoing ones that are going the characterization and the biological activities in cancer cell line. Thank you for listening.
Thank you so much for this beautiful work. The area of concern is cancer, and is area that we need to buckle up to find therapy. And I'm happy this one is coming up, and I hope that by the time you characterize, you're also going to go into, making it available. We can start with, it must be the isolate. Once it's very promising, you can also do, go into the formulation of the extracts if it's highly effective. So we mustn't wait for the isolate to characterize the compound and other things. So I thank you so much for the presentation, and I encourage you to get to the final stage of this work. Questions from audience? Okay.
Thank you very much for the presentation. I would like to make a comment on what the professor said. Yes, it's good that when you have activities like this, you can go as well ahead for maybe formulations. But again, you may need to consider the toxicity first. Okay? Yes, so you may need to carry out active toxicity and possibly subchronic toxicity of the sample. Yes, most anti-cancer agents, they have cytotoxic effects. But the cytotoxicity have to be different from that of the human cell. And that is why it is very important for you to do that. Now, I wanted to check your slide of isolation, those slides. If I can see one of them. Yeah, this one, right. I'm looking at this, the plates, 1, 2, 3. At the plate 1, I'm looking at about 4 spots. They're very close to the margin, the upper margin. Yeah. So what are they really? Now, because if you have places like this in real isolation work, it means that if you extend the length of this plate, you may have some other compounds there, and it is not really regular for us to have, something like that. Maybe your spot should be in the middle of the plate. So you may think of that going forward. Then the last plate, this should be 365 nanometer, right? I'm very sure. 365, right?
The first one is 254. Why the second one is 365?
The last one, I mean the last one. Yes. No. Yes, right. Okay, now, you see the problem with this is this. Most of these compounds here may not be real. They may just be, some chlorophyll and anthocyanins and the rest of them. So you may have to be very careful actually listing these ones as possible compounds you are going to isolate, because it's going to take a lot of time to be able to separate these compounds you are seeing there, because they are not really real compounds. Take care of that. Thank you.
Thank you, sir. So, ma'am, please, can I ask you? So, thank you very much, sir, for the question. As for the first question with regards to the length, I should start with this. I should start with the last one. This plate, this is the same plate we have here. Of course, we know that there are some constituents that are not chromogenic, that are not photogenic, that will not react to it, so you won't see on that microscope. So we have to do the one we spray with sulfuric acid, and which is this particular plate. And you can see that because initially we consider what we said, that it's possible that this is not real. In fact, my supervisor does not really take results from UV. So he asked me to repeat that for H2 and so forth, and that's why we have it. And as for the constituents up towards the front, they were fats. They were fats and impurity. So we did not really consider those ones. And can't remember any other question again?
Okay, let me just come in here. You see this your plates, right?
Yes.
It's showing us that these compounds are available here. But I believe that while the work is going on, you are at this junction, you have achieved one thing. These are the number of compounds present. And as he said, the one going up means that maybe they will still further separate as you move on, if you are giving it enough space. And I know that at this junction, what you have supposed to have done, which I believe you have done if you have sent the compounds, is that you now from here started the separation to get each of them. And we expect to see what you have placed here is also showing us the TLC plates for each of these compounds, showing us and the solvent mixtures used, showing us the first TLC and the 2D TLC. Because even when you get a TLC plate showing you a single spot, you also do a tilt of 90 degrees, that is 2D. You now show us move again. If there's anyone hiding or lapping, it will separate. So after that, then what you do is you now subject it not only UV. You can do iodine vapors, exposure. You can also do melting points. These are the things. So maybe because of space, I don't know, but these are the things. So this here, this chromatogram is just showing us they are present. I believe by the time you end to getting them sent out, you must have done these things, to get a pure compound standing alone, because they are together here. Do you understand? All right.
Thank you very much.
Okay.
A lot has been done, and it's because of the slide and the size.
Okay.
Just to give us appetizer.
Yeah. Okay, like what you are seeing here. These ones are single now.
Yes.
That's what I wanted to say. So you didn't even, you have better to answer your question. They're supposed to be in this form, not in that cluster, right?
Those are.
And you're supposed to have shown us weight, the mixtures, solvent mixture strength for these ones.
They are there.
Okay, maybe we are not at the top. So I think that's less better. Any other question? Okay. Yeah. Okay.
Well, thank you very much.
Yeah.
I'm sorry if I'm talking from the angle of being a supervisor. Apologies, please.
That's why we are here.
Of course, the results he presented right from the word go, it's more or less from the topic, bioactivity guided isolation. So those ones that we felt were not active were left out. So along the line, the major spots that we had eventually were the ones that we are using. Around from the word go, we don't use a specific spray reagent. We use the general one, H2SO4. And towards the end, we also try to check the effect of the iodine vapor. One striking thing here, I was just telling my brother here, there was one of them that overlapped in terms of the spots. It's like, if they use example 0.45 as RN value, the other one 0.47. So you can see the overlapping of the circles. So with that one, we had to use double prep TLC. Prep double in the sense that you want such material to have the length of the plate. Then you remove it. You won't spray it at that level. Then dry it and then dip it in the same solvent again. So the level, the origin now will now shift to where it stopped from, and where it stopped. So when we did that, there was a gap between the two of them. So through that, we were able to cut it off. And there were two compounds that we eventually obtain from it. So that's how we did some of them. So the way they are, just as we know that we don't have anywhere in the country where we do anti-cancer screening, the samples have been sent to Pakistan through our friend in Ilori, Dr. Salawu. So we've sent them to Pakistan, expecting their results. Thank you very much.
Yeah, thank you, DBC. That's a good work, as I've said earlier. So this, what we are seeing here is how a pure compound should look like, and there's the chromatogram, and you do the melting point and other studies that are. So well done so much. I think in absence of any other question, we go to the next presenter.
Good afternoon, everyone. I'm Dr. Rabueze, University of Lagos. Starting on existing protocol, I will be representing my team, presenting our work titled Drug Discovery, Isolation of Neuroprotective by Active Secondary Metabolites from Olas Osteopenia, root back a preliminary study. This will act like a guide for the presentation. Natural Products has done a lot. In fact, he has taken us a lot of milestones. You can imagine what penicillin and insulin have done in treatment outcome. But all the same, there are still areas that we are still battling with, something like neurodegenerative diseases. The memory loss, so much still needs to be done, because as of now, most of the drugs in the market are they deal with the symptoms and not with the underlying diseases. I came across this plant, Olax Osteopenia, when I did a botanical survey of the plant used for oral-dental infection. The traditional person, healer that gave it to me said it is used for psychosis, and that is actually what got my interest in. And that was in the Nsuka area. They call it Aziza. So the pictures we have here are the pictures of the different morphological parts of the plant. And the lower part of it, the figure 2, are some of the works we have done on this particular plant, because it's a continuous project. It has a good antimicrobial activity, especially fungi, oral fungi. And then the partial there is the individual neurodegenerative activity done using animal plant. So for the part of this project we did and presenting, the aim is to isolate cognitive improving by active compounds from hydrometallic methanolic fractions of Olas Osteopenia loop back. We did the phytochemical analysis for standardization, extraction using 80% methanol maceration. For the fractionation, we fractioned alkaloids and non-alkaloids using the normal procedure of dilute acid, acidifying with ammonia, and then using fractionating chloroform. But we discovered that the yield wasn't so much, so we used an alternative of ethyl acetate. And we subjected them, two of them, both the chloroform and the ethyl acetate fractions of non-alkaloid and alkaloid, to acetylcholinesterase inhibitory acid, which is an in vitro method for determining the effect on the cognitive dysfunction. And then we subjected one of them to activity-guided isolation and characterization. Using open color chromatography, purification was done with silica gel and then sulfadex, employing different types of elution method, isopropyl and gradient results. This just simply, this is not just a crude extract, that's a mistake. These are the crude extract, the various fractions we got. So the table 1 is the anticholinesterase activities of the various fractions. You notice the fractions we got from the chloroform, the alkaloid and non-alkaloid. The fractions we got using ethyl acetate to get our alkaloidal and non-alkaloidal. The activity shows that one, the yield, though it's insignificant, but we have more yield with ethyl acetate. And the activity shows using, let's say, 0.12 milligram per ml, that the activity of the non-alkaloidal fraction of chloroform and that of ethyl acetate alkaloidal fraction. The alkaloidal fraction of ethyl acetate gave more activity. But then that could be easily explained because ethyl acetate is used to extract flavonoids. So it's combined probably alkaloids and flavonoids. And that informs our using this particular fraction for our isolation. We were able to get 9 isolates, and so far we have been able to demonstrate 6 of them. 4 xanthone, 1 tritepine, and I think 1 flavonoid. So this one is you can notice that it's still coded because we are still working on the name, but using the 2D and the 1D NMR, we were able to get this, at least 6 of them so far, but we are still working, work in progress. And the isolates were subjected to its anticholinesterase activity, and then compared with the standard eserine, that's a physostigmine, normally used for management of memory loss. You notice that 6F, 16Y, 15Y, 14Y, they have we can classify them as moderate active. Okay. Now I'm going to single one of the compounds that we coded as orabase 15Y, which will represent others, or just let me just use it as a case study. We did the ethyl acetate alkaloidal fraction, like I said, was the one we put in the column. We did a gradient elution starting from 100% exempt to about 70-30 of ethyl acetate and methanol. We got 8 subfractions. And then subjecting it to acetylcholinesterase, we now pick the subfraction 3, and that is the one we now subjected to sulfadex using methanol. And then the fractions you see here, 1 to 32, they are not, we didn't put them together because we're collecting the 2 mils, 2 or 5 mils we're collecting. And we were looking for the most pure one, so we didn't put it together. So that's why I picked 14, 15, 16Y. They were subjected to NMR. And of all of them, 14 showed a good, the NMR shows it's clean, and that, but the 15 was the most cleanest and the pure of it. And then we used we were able to, from the structure, we were able to propose a formula, the C14H10C5, oxygen 5. In conclusion, our journey with Pharmacognosy has shown that actually it contains alkaloid, and the activity is not just in alkaloidal fraction. The activity, the crude extra showed activity, the alkaloidal fraction showed activity, and non-alkaloidal showed activity. both in the chloroform fraction and the ethyl acetate fraction. But that of ethyl acetate gave a higher instant, higher activity, showing that it contained more than chloroform, alkaloids, because ethyl acetate to extract flavonoids. So based on our results, 15Y, 14Y, and 6Y are the same. But they both pure, and that's why we went with this, say 15Y. They can still be, and it shows a good activity compared to the X-ray, that's the positive control. Well, it can be optimized through molecular docking to understand the mechanism, to understand it could lead to drug development if or subjecting it to molecular docking or AI training. These are my references, and then I acknowledge funders, MS, RFC, and the University of Oxford, where we did under the ASCOF initiative fellowship, where I did the MMR, the 2D and the 1D. And then this, I think, it's not showing, the last slide is not showing. That's the last slide where I have the GLCs of some of these.
Yeah, it's showing there.
Okay. The GLC there is that of 14 and 15, you can see there. So we didn't combine, we just picked that last one. And then this, I'm sorry I'm using my hand, is the 15Y. Anti-drug is a yellow powder. I think that's that.
A round of applause. Thank you so much, Ma, for your wonderful presentation. We now welcome questions from the audience.
Yeah. Well, thank you very much. Nice job. But I have some questions. Your first TSC plate, can I see it, please? The very first one. And even this last one too. Yes, right. Now, I want to believe this is not a pure compound, right?
No, this is the crude and the fractions. One from the alkaloid and non-alkaloid from the alkaloid from the chloroform and alkaloid from the fractions from the ethyl acetate.
Okay, so
just to confirm the presence
where
of
is the TSC plate of a pure compounds?
It's only one that I showed.
Where is it?
This one.
No, this is not pure.
So the TL.
This is not pure.
Can I expand the
Explain.
NMR?
Explain.
No, expand the NMR because
you can expand. You can expand, but from the TSC, you don't even need to expand NMR. We discussed it before, almost 2 times here. You run double TSC, and even you run how do you call it? Yeah, right. And because maybe the center you work at Oxford, I also work in that center too. You understand? But this compound you are claiming that is pure, it's not pure. I'm seeing it from here, and there are a whole lot of spots. By the time you change the solvent system, you'll be surprised that this all will pull out. So you cannot, if you subject this to NMR study, especially proto-NMRO, you are going to have a lot of, peaks.
It's here. Because I did it with several solvent systems.
I'm not going to believe what is there. I'll believe this TSC. Do you understand? Yeah, it's true. There are a lot of scientists here who have done this before.
I know.
Yeah, right.
What I'm saying is that I have the proton and the carbon 13. I have them here. But that we can
Sorry, show us the TSC of the products and of the carbon 13 and all these things. Can we have them, please?
I don't have them here. Because the work is still, the work has not been. That's why it's still coded.
Well done, madam. Your work is appreciated as well as the presentation. I just wanted to ask, with regards to your pure compounds, how many solvent systems did you use for the TSC of your isolated compounds?
For me, for us to try normal try.
Solvent system, and you got to one spot.
Yes. In fact, when Dr. Nick did the first, it was like, wow. That even here, when he did the first, because normally I did it first, normally they were subjected to 400 before they now take it to the one they use computer to see the purity. I know that it drops a lot.
Man, I'm not asking about the NMRO analysis. I just asked, when you do, of course, at the end point of your isolation process and you allude to the fact that you have a pure compound, the next thing you want to do is to try out different solvent systems to be sure that you still have your one spot. Because the what we found in most cases is that under one solvent system, if you are alluding to the fact that you have a pure compound, you'll be surprised that when you use another solvent system, you may have up to 2 or 3 spots as the case may be. So that's why I asked that question, to be sure that we are on the same page, especially if you are giving us this plate as a plate of your isolated compounds. All the same, thank you for your presentation.
All right. Let me. Okay. I said we read about 3. I've been trying to. Maybe you can help me to get where I have a proton NMR.
Okay. No.
No, it's only you see the number of.
Madam, wait, brother. We have all done some work or similar work like this. Before you proceed to doing NMRO, you want to be sure that your compound is pure. Now, the TLC you are showing us does not prove that. Number 2, you saw what the earlier presenter that presented before you. You saw by the time he got his pure compound, he now individually ran them as a single spot to be on it differently, individually. But what you are showing us is still congested together, and it's obvious that it has not been separated. So we are not doubting you. I know that you might probably want to quote some things because your work you have not gotten to the time of unveiling.
Actually, I said we are trying to trace something. And if you look at that, and try the TLC of the last one, there are different fractions collected and all that. There are different fractions. You see that of 14, 15, that one was, and then the other ones are different.
Okay, madam.
I didn't do anything on this. It's on this I did, and I did about 3 solvent systems. And if we can, after the presentation, you look at the proton.
Okay, I think you see, when you are packaging your presentation, you try to remove things that are ambiguous. No, you started that by telling us that the TLC chromatogram of your sample. So that's what we are looking at. We don't judge what we don't see, right? So we're just saying that if this is your pure compound, it is not. It's a step. It's one of the stages. So we had expected you to show us a chromatogram containing, wait now, I'm looking at it very well. I'm good at this. There are so many things there, right? So we don't want you to, I want you to tell us that this is not a pure compound. Simple. We move on because it can't be. And I believe from what you are saying, you have done so many to get those pure compounds separately. And I want to believe so. So, any other question? Okay, all right. So thank you, ma'am, and we are so grateful for your presentation. Thank you so much.
Hello. No one tell us how you or you didn't capture it.
Thank you very much for the
For
last
every 2
sorry.
Hello, the session is still on. Please come continue the discussion later.
Sorry, ma'am. I just want to say
Okay,
something.
okay.
You know, natural product chemistry is not a very easy thing.
Yeah.
And we all know that, you know, especially pure or not pure, if it's not pure, you actually will get it easily from the NMR too. And I think you should clearly. So I think the summary does that this work is still in progress. So I think there's a lot of room to continue. I wanted to find out from her. She showed some, I think crystals.
No, the powder.
Yeah.
That one, that 15 white.
Yeah.
14, 15, 16. Within that
Was it
one, that one white. They were powder.
Was it the one that took the NMR?
Eh?
Was it the one that took the NMR?
Yes, I did NMR for 14 white.
Powder that you showed?
Yeah. The yellow powder.
Yes.
Even that yellow powder, even before I did it, I still ran it through several times.
Is it the one who did the NMR?
Yes, one of them.
And you said it is pure?
One of them, yes.
Okay.
I ran it through
I wanted to find out, was the melting point of that?
No, I have not done the melting point.
Not done yet?
Yes.
Thank you. Because even for the melting point, you can say no.
Madam, did you take the melting point?
Yeah.
You've not done the melting point. So there's no way you will know that the compound is pure even when you are not determining the melting point. If you take the melting point, you will see it's going to be broad. It's going to be wide. It won't be
Okay, can we see the slide? Okay, Bello. Who is presenting? Bello? Okay. Bello and
I'm Ronke Hadia Bello, presenting my abstract topic titled Antibacterial Efficacy of Eugenia carophilus and Osimum basilicum essential oil against selected multi-drug resistant uropathogenic E. coli. Urinary tract infection is an infection of the urinary tract system, whether it is invaded by microorganisms. One of the most commonly occurring etiologic agents of this infection is E. coli. The emergence of multi-drug resistant uropathogenic E. coli, particularly those that are resistant to the penicillin as well as the cephalosporins, has been documented. More recently, the quinolone resistance, cold resistance has been reported. So this necessitates effective therapeutic options. Clove and sweet basil essential oil have demonstrated several antimicrobial resistance to different antimicrobial activity to different microorganisms. This study will generate scientific evidence for the use of this clove and essential oil and sweet basil essential oil as plant-derived antimicrobial agents against multi-drug resistant uropathogenic E. coli, offering promising alternative and complementary therapeutic strategy. To this, the aim of this is to evaluate the antibacterial efficacy of this selected essential oil against multi-drug resistant uropathogenic E. coli. Figure 1 here shows the flowchart of our sequence of activity. The multi-drug resistant uropathogenic E. coli was collected from the work that was previously reported by Bello et al. And this was screened against the sweet basil and clove essential oil. We also tried to look at their MIC determination, and lastly, to determine their synergistic effect using the checkerboard method. These multi-drug resistant E. coli were the result and discussion. Multi-drug resistant E. coli were tested for ESBL producing and quinolone resistant using the disc diffusion method as described by CLSI. 19% of our isolates exhibited co-resistance to ESBL and quinolone. And they exhibited different degree of inhibition against the selected essential oil, with sweet basil showing the high zone of inhibition. With the mean MIC of 2.5 to 10% MIC for both oils, and table 1 here shows the checkerboard summary of our analysis, with about 60% of our isolates showing synergistic active effect, and with the mean
0.7 was obtained, showing an additive overall effect. This additive effect, which is still beneficial, and suggests that this essential oil could be formulated into effective formulation. However, this work is in progress. We are still carrying out some other analysis, formulation, as well as some pharmacological analysis will still be carried out. Finally, in conclusion, essential oil of these selected essential oils of clove and sweet basil can be used as alternative to overcome challenges of these multi-drug resistant uropathogenic E. coli. However, with the documentation of safety of use. These are some of the references that we use. Thank you for listening.
Thank you so much for your presentation, Bello, for your very timely within the time. Please, we need questions from the audience. Do we have any question from the audience? Okay, can we go to your results? So what's the plate what? Because we can't see, this one is not working. That plate 2, the one below the figure.
The plate 1 showed the pure isolate of the E. coli that was collected.
The what?
The pure isolate of E. coli that was collected.
Okay.
Then plate 2 showed the screening for the ESBL detection and quinolone resistance among the isolates.
They'll have 2 plates there.
Yes, one is for ESBL detection and the other one is for the quinolone resistance.
Okay. Then the other plate 3
was the screening process for the antibacterial activity of the essential oils.
So what can you say from the plate 3 that's A and B of it, so that we can understand?
Okay, the A and B of it at different concentration we tested for activity. So at the highest concentration, which was 10% volume in volume, that was where we had activity. And since it was showing promising activity here, we ran the MIC using the broad dilution because we were looking at the agar, maybe it was contributing to the non-pronounced inhibition. So at the broad level, it was more pronounced.
Okay, thank you so much. Any other question from the audience? Okay.
Please, I just wanted to find out if the oils are combined or separate.
Yes, what we did initially at the figure 3, it was single. So we did the antibacterial susceptibility for the oils individually. But here, at the checkerboard level, we combined. So that's where we were able to look at the FIC and see their combinatory effect.
Okay, thank you so much. Thank you so much, Bello. Okay, hold on.
Very interesting work here, and I appreciate the clarity of the presentation, sharp, straight to the point. The graph before this, where you have the 3-bar chart, I want to know exactly, because from here I can't see what this represents. The big red bar, sorry I have to use that, and the one that is just at the point, what do those represent?
Okay, the first bar chart there, we were trying to look at the resistance profile. We had 30 of our isolates that were ESBL producers only. Then the quinolone resistant, none of the isolates were quinolone resistant. Then we have 20% of our isolates had co-resistance to the cephalosporins as well as the quinolones. So for the second bar chart there, we try to look at the zones of inhibition to the selected essential oils.
They are clinical isolates?
Yes, they are uropathogenic E. coli.
All right, thank you. I just needed clarity.
Okay, round of applause. The next presenter, please. If the next presenter is available, the person is here, please. Let's. Oh, you are the presenter. Oh. Okay, that's fine. That's fine.
Good afternoon, everyone. Good afternoon. My name is Ezekiel Efoka. I'm here to present on the acaricida properties of Ageratum condyzoides, Sipupogon citratus, Latana camara, and Praxelis clematidae. And this work is a joint effort by my supervisor and my former HOD and some other lecturers in other departments in the University of Benin. This is the outline of my presentation. And as an introduction, we have ticks, major vectors of disease. These diseases, sometimes when they are passed to animals, are usually transferred to humans upon when they are harvested or when they are processed for eating. And these ticks sometimes can also attach themselves to humans and then transmit illnesses of different varieties. So the aim of this presentation is to ensure that we look for natural acaricida agents that can help kill off these ticks and then also reduce the effect of the initial methods that we have, that is chemical control. Chemical controls are known to cause severe side effects. Then we also have physical control. Physical control of ticks, we find situations where the cattle rearers, which are they go physically to unpick these ticks from the cattle, and these are quite laborious and difficult. Then there is also the bush burning. Bush burning where they actually, because of the infestation of ticks, the cattle rearers set up a particular part of the forest where the cattle have been staying for a while, and then move the cattle to a different location because of ticks infestation. I know how that has been a very major concern for security and economy in this country. So having a natural acaricide is actually beneficial to both the health of the cattle, the economic value of the cattle, and the financial or economic effect on the cattle rearers themselves. And that is the justification for this study. So samples of the plants were collected from University of Benin, and they were authenticated in the Department of Plant Biotechnology. And these are samples and pictures of the plants. These are samples and pictures of Latana camara and Praxelis clematidae. So the volatile oils of these plants were extracted using hydrodistillation, where with heat, the oils that are present in this plant are then collected and then condensed immediately over a collector from the condenser. The ticks were collected from the cattle, and they were also authenticated in the veterinary department in university. They were shared into different species, and those species were selected into 10 groups each in 4 different petri dishes. It is in those collections where we applied the volatile oils gotten from these plants. So this is just a short display of how the ticks were harvested from the animals. It's quite very laborious, and sometimes when you meet these cattle rearers to collect these ticks, they are not very supportive, except we stimulate them a little bit because it's quite a tedious process. Because of time, we'll pause the video there. Sorry. All right. Then the oils were applied. The oils were applied at different concentrations: 1%, 2%, 5%, and 10% on the different ticks group that were selected. And these are the results. Different tick species selected into their different ticks. Rhipicephalus microplus, which is designated big ticks, and the Hyaloma rufipis, which is designated as small ticks on the result here. And this is the results of the effect of volatile oils on these ticks. Now, as we can see that as at 1%, that these oils already have a considerable number of effects on these ticks. Worthy of note is Sipupogon citratus, which is commonly known as lemongrass. Lemongrass had a 100% mortality rate for these ticks as just at low percent. We recorded 100% mortality for all the volatile oils as high as 10% concentrations of the volatile oil. So in conclusion, the effect of this volatile oil has shown that this is a very promising aspect of research. We have actually formulated this oil and taken it for analysis. We actually worked on different fractions, and the oils were formulated and then applied on the cattle ticks in the cattle on the cattle farms, and we're able to record our results there. Thank you very much.
strengths in the PowerPoint. How do I put it? Very good. They are very good at PowerPoint. That's for PowerPoint skills. When I was watching the short video of you blowing it, I was wondering, are you the person of these people headers? Okay, so that's a good one. The presentation was on time, and I thank you so much for that. Questions from the audience?
Yeah, thank you, Ezekiel, for the wonderful presentation. I wanted to ask, these are volatile oils, right? We work on volatile oils, right? But I wanted to know, because it is a little bit difficult, really, and time-consuming to extract volatile oil, and then also be able to use them on the treatment of cattle. Okay? How do you intend to upscale or optimize these extraction methods so that you can have enough volume of volatile oil for the treatment of a herd, of maybe about 1,000 cattle? How do you intend to do that? Thank you.
Thank you so much for your question. So we are listening.
Okay, thank you very much for the question, sir. I think the percentage concentration of the oils we found that were effective, we found percentage as low as 1%. And in developed worlds, we find that these cattle are placed in ranches, and they have a gutter system where the cattle walk in, where they dip themselves into a marshmallows of water and apply whatever chemicals they want to apply. Usually, when they come back from moving around, they lead them through that ditch. They walk in and then come out, and whatever oils that is needed to be applied is already applied on their skin. However, because most times what we find in Nigeria is that we find a group of cattle owners coming together to keep their cattle together. So we could not actually get them together to agree to build something like that. So we manually apply these oils, the consumption of the oils on their skin. However, if they are going to use this as an acaricider agent for cattle, I think we'll have to use that method where we make something like that, have the concentration of the water or the solvent there, perfect for the cattle, and we'll allow them walk through. And that will solve the problem of application.
Did your question answered? All right, okay. Is there any question from the audience? I was also thinking why you should also, maybe the work is ongoing, you can also run the GCMS analysis. Okay, you have done that. Okay, send it for, okay, that's fine, that's fine. Any other questions? Okay, just hold on.
Have you identified, I'm not talking of from TCMS, were you able to characterize any compound from it?
Yes, we were able to. From particularly Praxelis clematica.
What's the name?
We were able to identify a pure compound, but we did not characterize.
It's not characterized.
Okay, thank you so much.
Okay, my question is, how do you get a very large quantity of this oil? Because this method of extracting this oil is usually very tedious and small. So how do you maximize it?
I've been doing this project for the past 10 years.
Jesus.
You said what?
He has been on this job for 3 years.
Wow, that's lovely. That's interesting.
So you now know.
That's interesting. That's interesting. Okay, well done. We appreciate that. A round of applause, Jim. Okay, the next presenter.
Nelson.
Okay, you want to ask a question? Okay, okay.
Please, did you combine this oil?
Hold on.
Just seeing it.
Individually.
Individually.
for yes, maybe, probably, I don't know. My supervisor is an answer.
It's not for your supervisor. Don't worry, sir. Okay, you. Are you not going to answer?
No, it's not part of his work.
Okay, it's not part of.
Supervisor want to.
Okay, it's all right. Please, a round of applause again. Yeah. All right. The next presenter is it Imeje or Saib? Who is presenting? What's your name? Saib. Okay, you're welcome.
Thank you. Good afternoon, students. My name is presenting on the topic. Good afternoon, esteemed guests. Standing on the existing protocol, my name is Nelson and I will be presenting on the topic in silico evaluation of GCMS identified and isolated compounds from Annona muricata as possible potential inhibitors of SARS-CoV-2 main proteins. These studies was carried out in the Department of Pharmaceutical Chemistry under the supervision of Dr. Vincent Nigerian. Okay, as we know, COVID-19 caused a global pandemic as well as a health and mortality crisis, and there was a need for sourcing of new antiviral agents. And one of such antiviral agents is known as remdesivir, although it comes with its side effects, which is a very toxicity profile. So there is a need to source for new antivirals, and one of such new approaches we could use is computer-aided drug design, which is CAD. So computer-aided drug design helps us to virtually screen bioactive compounds in medicinal plants. And one of these medicinal plants of concern in this research is Annona muricata. And Annona muricata is known to contain rich and bioactive compounds like acetogenes, alkaloids, flavonoids. Hence, the aim of this study is to evaluate the phytochemical constituent embedded in Annona muricata to see if they are potential inhibitors of SARS-CoV-2 proteins. The objectives of these studies were to identify compounds via GCMS, to dock the selected ligands against the SARS-CoV-2 main proteins and compare with remdesivir, which is the standard drug. Then also to evaluate the absorption, distribution, metabolism, and excretion, and the drug likeness using Swiss Army, and also to predict the toxicity using Protox 3 web server, and to identify safe and high affinity leads. Okay, so for the method, we obtained fresh leaves of Annona muricata from Kosodi District, Nigeria. And these plant leaves were identified and authenticated at the University of Benin, Liberia, by plant taxonomists with the herbarium number UPH8356. So the leaves were then air dried, they were pulverized into fine powders, and we extracted them using methanol for 7 days, after which we sent them for GCMS analysis, and we got the identified phytochemicals. And these identified phytochemicals, alongside isolated compounds we obtained from literature, we are then retrieved from PubChem server. We minimized the energy and we converted it to .pdbqt, which is in appropriate molecular docking format. After which we obtained the protein target of choice here, which is the main protease, which is 6NU7, which is the ID for the protein target. After obtaining it, we used the biovia discovery. We cleaned it, we removed water, we removed the heteroatoms, and we removed the co-crystallized ligand. Then we also added hydrogen bonds to enhance the stability. After doing that, we went on to carry out the docking, which we used the Pyrex software. In Pyrex software, we have an auto-dock cleaner, which is a tool used for the docking. We docked. After docking, we took it to biovia discovery again to visualize the molecular interactions, which are in 2 dimensions and also 3 dimensions. And we also compared those that had less than -7.5 and selected them for ADME as well as toxicity studies. As you can see from the results, we only had about one that was put there for representative of the identified phytochemicals, although we didn't have good binding affinity, -5.8. But from number 2 to number 8, we are seeing very beautiful binding affinities from -7.7, -7.8, -8.8, and the highest being quercetin 3-rubinoside. And we can see it has a very beautiful binding affinity, much better than remdesivir, which is the positive control. Then we can see the Lipinski's rule, rule of 5, the number of violations, we have from 0, 1, 2, and 3. Although the Urinde survey has 2, the quantitative revenue side, which is the highest, has 3. Though that's a drawback, but we could also modify. Then toxicity, we can see there are different toxicity class, 5, 4, with 5 being the best. Then, as you can see for quantitative revenue side, beating that of Urinde survey for class 4. So, like we said, in several another Moricata phytochemicals, they obviously show strong binding affinity against the SARS-CoV-2 main protease, such as those maurine, camphorol, quercetin. So we know that they have good oral bioavailability as well. Then for flavonoid glycosides, such as the quercetin, like we said, they show stronger, like I mentioned. But overall, we know that the another Moricata possess rich and bioactive compounds that gives us very safe ADMET profile as well as the potential leads or promising leads. So in conclusion, phytochemical as well as molecular docking analysis confirm that in Anona moricata, we have promising leads that serve as potential inhibitors against SARS-CoV-2 main proteins, although it is essential to carry out further in vitro and in vivo studies to validate these claims. Then for my acknowledgement, I would like to acknowledge the National Product Research Lab, University of Benin City, and also TED Fund for their available support towards this research. These are my references. Thank you.
A round of applause. Thank you, Saibu, for the well-packaged presentation, and also you are timely, and we appreciate that. I want to make a comment on the title. Because if you look at the title, someone will be thinking that you isolated compounds, right? So we have to make an adjustment to that, because you are referring to, those ones previously, it's identified GCMS, not isolated. Okay, yeah, so that's where I was waiting to see the next the processes. So it just put identified compounds for. Thank you very much.
Thank you.
Question? Okay.
Not really a question. I want to, while he was doing his presentation, I had to ask whether he's a postgraduate student, but I've been told he's just finished his FMD program.
Wow.
And it's an excellent presentation that you have done today. So I'm really very hopeful that you will not mind the poor remuneration of your lecturers. They will want you to come back and continue. Thank you for a beautiful presentation.
Thank you for finding out. Very, very useful. So you're just a first degree. Oh, you are not finished. Oh, you're still doing your project. Jesus Christ. This is wonderful.
Pharmaceutical chemistry.
Wow! You know this professor here, you have a good student. Don't let him go. He will soon jump back. Automatic NSP member. I'm telling you, automatic. Okay, our BBC is here. See, it's now in your custody. I will be asking you of this guy. Thank you so much for that excellent presentation. A round of applause. So the next presenter, you are the next presenter, Ezekiel? Okay, all right.
Good afternoon, esteemed guests. My name is Amdele Ayedodo. I'm here to actually make a presentation on my undergraduate session titled In Silico Antiplasmodial Evaluation and In Vivo Acute Toxicity Assessment of Combined Acroleal Cordifolia and Inertia Chlorantha Extract
against Plasmodium falciparum plasmacin 2. This work was not done by my humble self alone. My project supervisor, Dr. Emeji, actually played a massive role towards this project. And for that, sir, I say thank you very much, sir. Yeah, to the background of the study, malaria, the burden of malaria. Malaria primarily caused by Plasmodium falciparum remains a major global health concern, worsened by drug-resistant strains. For the purpose of this study, our target is plasmapepsin 2. I would say it's hemoglobin degrading aspartic proteins that is crucial for parasite survival, thereby making it an attractive drug target. The aim of the study to identify and evaluate phytochemicals from Acordia cordifolia and Ennarcia chlorantha as potential anti-malaria targets, agents against P. falciparum plasmapepsin 2 with a PDB-ID 1-LEE through molecular docking, ADMET profiling, drug likeness assessment, and in vivo acute oral toxicity testing. Now to the methodology of this session. The plant collection, the fresh leaves of Acordia cordifolia and stem bark of Ennarcia chlorantha were harvested from Edo State, Nigeria. Their botanical identities were confirmed and authenticated by a plant taxonomist at the University of Benin. We provided voucher numbers. The preparation, the leaves and the stem barks were air-dried and pulverized into fine powders. Extraction and concentration, continuous solvent extraction was done using methanol for 7 days. Then we freeze-dried and concentrated using a rotary evaporator at 40 degrees Celsius. GCMS was also used to identify the phytochemicals. The identified phytochemicals were then retrieved from the PubChem database. They were minimized and converted to PDB-QT, which is the standard format for molecular docking. We now say for the potent target, the plasmapepsin 2 with PDB-1-LEE, which was also obtained from the protein data bank and prepared. It was prepared using Power Discovery Studio. Then our docking analysis. Our docking was done using Pyres. Using Pyres orthodox vena, and validated with a co-crystallized ligand R36. Interactions were visualized in Power Discovery Studio. Binding residues confirmed via PLIP, that is the protein ligand interaction profile of PLIP, and then PIMO compounds. And then compounds with binding affinity that were less than minus 7.0 kilocal per mole was chosen for ADMET toxicity. Acute oral toxicity testing, this was carried out using the LOX et al. Method. And just as we know, the LOX et al. Method is actually divided into 2 major phases. We have the phase 1 and then we have the phase 2. For the phase 1, what we did, we actually got about 9 mice and then divided them into various groups. And then we administered doses ranging from 1,000 milligram per kg to 1,000 milligram per kg. And then in phase 2, we gave 1,600 milligram per kg. We gave 2,900 milligram per kg and then 5,000 milligram per kg. And then for my results and for our results and discussion for the docking scores, or for highest binders for AC, looping on, we have looping on with minus 9.1 kilocal per mole, Friday 8.3 kilocal per mole, and beta-cystosterol. And then for our EC and Ennarcia chlorantha, we have the stigmasterol, berberine, and then we had to compare these binding affinities to our standard, which was dihydroartemisinin. And what we obtained, the binding affinity for our standard was minus 7.6 kilocal per mole. And then for drug likeness, alkaloid, berberine, and permethrin through pterolipinski evaluation and good oral power availability. And then for in vivo acute toxicity, no mortality or visible toxicity was observed at the maximum dose of 500 milligram per kg for the combined extract. And then as you can see, this is our 2D and 3D images that we obtained for the promising ligands. And then then for our conclusion, from this study we concluded that this study confirms that phytochemicals obtained from Acronia cordifolia and the natural Rwanda ester shows strong binding affinity for plasmid stool. And then the combined ester also demonstrated a high margin of safety. In vivo LD50 that was greater than 5,000 milligrams per kg. Acknowledgement, we gratefully acknowledge the test fund and the National Product Research Laboratory, Department of Pharmaceutical Chemistry, University of Benin, for their support and provision of research materials. References are actually being stated. Thank you very much for listening.
Thank you. A round of applause. Thank you for your presentation. Thank you for the presentation and timely too. Questions from the audience? Okay. This place is where you start.
All right, thank you very much for your presentation. Actually, you started with an acknowledgement. So I would have preferred that you acknowledge your supervisor's role and the acknowledgement after presentation. That is what is generally acceptable. And also, the quality of your slides, they were not very good. And then also, the presentation of your results, it wasn't catchy. It was like you just made them in words, and we find it difficult to follow you. So, learn from what others did, so that you become a pro in the near future. Thank you.
Thank you so much.
All right, thank you for your presentation. I was wondering, you know, before we go for molecular docking and all these studies, sometimes anti-malaria studies is something that is very vast in Africa, I know. And I was wondering, is Acronia cordifolia, where it has ever exhibited any anti-malaria activity reported? Because I was thinking that before you can go to start thinking of molecular docking, there could have been a kind of assay that has reported a clarity of these effectiveness of these plants.
Okay, sir.
Yes, Onatia glurantia is known, but Acronia cordifolia.
Okay, I hope you understood him. Yeah, there should be literature reference to the subjects of study and the activity you have studied. There should be literature on that. I know of Onatia glurantia. That has also, that one is, has history, or you can say literature. But the Acronia is where they need to have been a little, or do you have, because sometimes there might be information you have that the audience, yes, because we encourage people to go for particular plants based on certain information. Can be ethno value, maybe from people using it, traditional use. You have found it, reported maybe in other places through literature review, and maybe it is not in Nigeria or it's in North. You want to study from the South or East End, something like that. All right. So, do you have any such information?
Yes.
Okay.
In times past, from literature review, my supervisor has actually worked on the Acronia cordifolia for research, and not majorly on malaria per se, because the faculty work on Acronia cordifolia for this is not malaria.
So it's not.
It's not.
Okay.
What is
just wanted to try it on malaria.
It's also good because sometimes you can defend your approach. You're choosing that plant base, maybe it's belong to the same genus, or they're different species, right? So sometimes we can narrow our search for biotics from reference that this group of genus that they do possess this activity. So let me try this species. Okay. This was want to say something. Okay. Okay, it's there already. Okay. So Daddy was okay. It was only done that.
Yeah, they've already done that.
Thank you very much for the
Fine.
good work and of course blessing to this good work.
Yeah.
I just want to make a comment on your conclusion. You said the compounds obtained from the plants was what you used in the book. Is that correct?
Because I said the study confirmed that phytochemicals from Aquana portfolio and the national production show strong binding affinity.
Phytochemicals what?
Phytochemicals, yes, from Aquana portfolio.
I think if you say reported, because I don't think you actually obtained them. I think they were reported, isn't it? Already reported in literature.
Yes.
Am I correct? Yes. So be clear about that.
Okay, thank you so much for your presentation. We appreciate you. And you're also first degree student, right? Oh, we are so happy. A round of applause for you. That's wonderful presentation. Thank you so much. Okay. Okay, oh, thank you all. I learned that he is the last presenter, and I'm happy we have come to end of this physical presentation and also the last section of this technical session for today. So I want to appreciate all of us, our daddy and mentor, our Professor Emeritus and Fellow with us since yesterday. We are happy that you are still with us here. We appreciate you. Our distinguished and indefatigable Presido and the assessor here with me, our distinguished professor, and also Professor Oga that's not here, have left. I thank our technical team, our ICT team for job well done. Please a round of applause for them. And our DJ and others that contributed, the presenters, the participants, the online presenters and participants. We are so much grateful and we appreciate all of you. Thank you all. And as we move to the next stage of the program today, thank you all. I forgot to thank our chair, the LOC chair of this conference. You have done so well. A round of applause for her and her team. And the HOD of Pharmacognosy that will be moving up and down. Our Professor Abere and the rest of them. Please, we appreciate all of you. And other faculty members from different departments, we appreciate all of you. Thank you all. Our BBC is here. Bring your microphone before I'll start going with my legs. We have I didn't know that he is still here. So we thank our DVC from also a distinguished fellow, a mentor too, and our elder he is. We appreciate you. It's because of you that we are here. We're enjoying these days, 3 days, me in particular, I've been here since 3 days, and we're enjoying the breeze and environment of Benin. Courtesy of him and the team. Thank you so much. Thank you so much.
Before we go ahead, I was just telling him that for his presence, we want to particularly appreciate our DVC for light extended to this particular venue. Honestly, but for his intervention, light would have been cut off from this place. So our DVC, sir, we appreciate you. Thank you. Thank you. Thank you, sir. So shortly we'll be going to into the AGM. And before we go into the AGM, there are certain ground rules that we have to establish. And the first of it is that non-members are not supposed to be part of AGM. So if you are a member, you have not been inducted, and you want to become a member, so please just raise up your hand so that you can be part of the AGM. Thank you. So that you can be a part of the AGM, we don't want to send anybody out. Please, those who want to be... Where's my mama? Mama? Mama Lago, oh yeah, oh. You get plenty children.
Those who want prospective members, yes, they will sit together somewhere.
Yes.
All prospective
the NSP, and we come together. We don't want to send anybody out. You see what we have been doing. The NSP is a beautiful society of scientists.
Only music. Prospective members come and stay here. All other non-members of the we love you. As students, we love you. All other people love you. It has become you. Yes, prospective members. So, we are calling the AGS.
Come and sit down first. You will get prospective students. Come and sit down first. You will get instruction.
All members of the executive, please move to the high table. Members of the executive, Dr. Saigade, you are going to take the minutes for this. Please move to the high table. And all our fellows, they should also move to the high table. Give this to them. And there is just process. Move to the high table. Then you are rounding up. Yes. Our assessors, Dr. Man, you have the result. Yes. So you, yes. You will need to submit your report now. You can sit here.
Remove the.
Yes, you need to.
Let's go! Let's go! Let's go! Please, the instructions are very clear. If you are not a member or you don't become a prospective member, please excuse us. Thank you. Except the teacher and maybe the IC. Please. Please. Those are also known as Mr. Basisa. If you are not a prospective member or a member of NSP, can you excuse us? Thank you. Please, for our student participants, don't go too far. We'll take care of it. But this one that we are getting into is a session for NSP members. Do you understand, please? It's not as if you're here. Yes, yes. Please, can our assessors also join them at the high table? Our assessors also join them at the high table.
Yes, yes. AGM should be streamed online too. Yes. Because we're going to have online participants. Yes, the online participants. I want to complete some things.